This is the second half of the immune system part 1. In the first half of the last video, we talked about the granulocytes, the neutrophils, eosinophils, basophils, mast cells, monocytes, and the dendritic and macrophages that can be the bridge between the innate immune system and the adaptive immune system. In this video, the second half, we are continuing on with the lymphocytes, and those are the natural killer cells, the T cells, and the B cells. Lymphocytes are a type of white blood cell that plays a crucial role in the immune system, helping the body fight infections and diseases. They are primarily found in the blood, the lymphatic system, the lymphoid organs, which is the spleen, thymus, and lymph nodes. And the type of lymphocytes are B lymphocytes, which are B cells, and they produce antibodies to fight infections. They are involved in humoral immunity. And remember, humoral immunity or response are B cells that produce antigens to help fight off the infection, foreign object, or disease. Unlike T cells, who completely destroy the entire cell, B cells only try to kill the pathogen and saving the cell it's in. T cells are involved in cell-mediated immunity. And cellular immunity, or cellular response, like T cells, hunt down and kill the infected diseased cell through a process called apoptosis. Think of it as a kill 'em all and take no prisoners approach. This process is known as cellular immunity, meaning it just completely kills off the entire cell. When chemical signals bind to their corresponding receptors, it induces events within the cell that ultimately changes its behavior. And you'll start to see how this fits in as we go through the immune system. We looked at this in the first half of the video, involving white blood cells and how they reach out with their little arms to the surrounding cell. They were telling the cell to go into apoptosis or self-destruction. The T cell works very similarly. There are several types of T cells. There's the helper T cells, and these are the CD4 plus T cells, for those of you that know whether you have a problem between your CD8 or your CD4. Your helper T cells are the CD4 plus, and they help activate other immune cells. Cytotoxic T cells, those are your CD8 plus, they kill the virus-infected or cancerous cells. And your regulatory T cells help maintain immune system balance and prevent autoimmunity. And now, natural killer cells is another form of lymphocytes. They destroy virus-infected cells and cancer cells. Natural killer cells are actually considered part of the innate immune system, because unlike the adaptive immune system, which requires prior exposure to a specific antigen, natural killer cells can rapidly recognize and destroy target cells without any prior notifications or training. Natural killer cells don't require the recognition of specific antigens through T cell receptors or B cell receptors like the cells of the immune system, adaptive immune system. However, they identify cells that have been altered on surface molecules, often due to viral infection or cancer's transformation, and then eliminate them. So basically, they can tell when a cell is damaged, either from infection, cancer, etc. They don't need any fancy signaling to get them going. They can do it all on their own without any help. They know when a cell is infected, damaged, or cancerous. And we'll be talking about this when we get to the natural killer section later on in this video. Okay, so like we said, lymphocytes, which are your B cells, T cells, natural killer cells, recognize and fight infections, viruses, bacteria, and pathogens. Also provide long-term immunity because they create memory cells. So that way the next time they come into contact with a pathogen, they remember it and know exactly what to do. They help regulate immune responses to prevent excessive reactions. They are produced in the bone marrow and move into the lymphatic system to protect you. Approximately 25% remain in the bone marrow and become B cells. The other 75% move into the lymphatic system and become T cells. There are two kinds of B and T cells. One is an effector cell, which is activated by antigens, so foreign proteins, to fight an active infection. and the other one is memory cells. These have been in your body long enough to recognize and remember past infections and go into action quickly if you become reinfected. Normal lymphocyte count is 1,000 to about, we'll say 5,000 cells per microliter of blood in adults. A high count, which is lymphocytosis, may indicate infection, inflammation, or certain types of blood cancers. A low count, which is lymphocytopenia, can result from the viral infections, autoimmune diseases, or immune suppression. The major histocompatibility complex. I actually tried to figure out the best way to go about teaching this, whether I should put it before or after we discuss the B cells and T cells. But the reason I need to teach you this is because it's how the B and T cells communicate and allows the T cells to recognize and respond to foreign material, triggering an immune response. It's what helps your immune system determine friend or foe. So this is what helps your immune tolerance, whether your immune system is going to attack you or just the foreign invaders. The major histocompatibility complex is a group of genes that code for proteins that help the immune system. These proteins are found on cell surfaces and are called the MHC complex. For example, a T cell is only able to recognize antigens if it's bound to an MHC molecule, unlike B cells that can respond directly to the antigen. So the MHC molecule is essential for T cells to work properly in the immune system. The MHC is highly polymorphic, meaning there are many different variants of each gene. The polymorphism helps protect against viruses that try to escape the immune system. Because they are highly polymorphic, this makes it very rare for two people to have the same set of MHC molecules. This makes organ transplantation more difficult, and this is part of all the testing you have to do to make sure you can have an organ transplant with someone else. MHC 1 and 2 are highly involved in deciding organ transplantation matches. It is also greatly involved in autoimmune diseases. And now you know why I absolutely have to talk about this. There is also another name for the major histocompatibility complex. It's the human leukocyte antigen. That's the HLA. And some of you may be aware of exactly what that is with your health conditions. And we'll be discussing a little bit about that shortly. There are actually 3 classes of the major histocompatibility complex. Class 1 is present on all nucleated cells except for mature red blood cells, which lack a nucleus. They present antigens to T cells, the cytotoxic CD8+ cells, as well as macrophages, dendritic cells, and even platelets, but the main purpose is to activate those cytotoxic T cells. Major histocompatibility complex class 2 is primarily found on antigen-presenting cells like dendritic, macrophages, and B cells, presenting the antigens again to the T cells. So the dendritic, macrophages, B cells present to the T cells using the MHC class 2. MHC class 3 do not present antigens. Instead, they are triggered by inflammation signals inside the cells and the cytokines that get released. And here you might start to understand that this is why I need to talk about this, because it also triggers mast cells and the other cells as well. And this has a very close relationship with your microbiome, your mitochondria, and your methylation. The function of the MHC molecules is to present peptides to T cells. This is where the T cells kind of get all murder-like. When it comes to pathogenic proteins, etc., they take the nuclear option. Kill all, take no prisoners. So the MHC class 1 molecules present peptides to the killer T cells. So killer T cells are also called cytotoxic T cells. Killer T cells, or cytotoxic T cells, can only bind to MHC class 1. They wait to collect their antigen. These are found inside the cell, and the MHC chops up the dangerous protein and then presents it to the killer T cell for utter destruction and annihilation. Pathogens are generally found inside the cells. MHC class II molecules present protein peptides to helper T cells. That's your CD4+. CD4+ or helper T cells can only bind to MHC class II. They wait to collect their antigen. They are wrapped in a bow called a phagosome to contain them, and then present them to the CD4+ T cell through various processes. To illustrate this, I found the perfect video to help you to understand what it does.
For this sketch, we're in the castle kitchens, where we're going to do a high-low thing, like an old English period piece, because there's nothing more topical than Downton Abbey. But seriously, splitting the sketch in half is going to help you keep MHC1 and MHC2 organized in your mind. Then it turns out that preparing dragon meat for knights and squires involves some important steps, and these steps will help you remember how antigens are processed and presented to T cells. We'll start with some general setup. First off, you'll note that access to these kitchens is restricted, a reminder of MHC restriction, because a T cell is only able to recognize an antigen if it's bound to a specific MHC molecule. This is in contrast to B cells, which are able to respond directly to an antigen. Getting more specific, CD8 positive T cells always only bind to MHC class I, hence our killer knight waiting to collect their antigen, or dinner, on the first floor. Helper squires, CD4 positive T cells, have to climb to the second floor to collect their supper, as CD4 positive T cells can only bind to MHC class II. And maybe you've already heard this a million times, but another way to remember this is that if you multiply the CD number and the MHC class, it should equal 8. 1 times 8, or 2 times 4. Alright, these hungry T cells are waiting on dinner, so let's get cooking. The overall goal of this castle kitchen is to break down antigens and load them onto MHC molecules so that they can be served to the proper T cell. Let's start on the first floor, or with the MHC1 pathway. So, it's kind of gross, but this bottom kitchen is infested by little mouse dragons that live inside the kitchen. That's not the gross part. The gross part is that they taste like chicken. Anyway, these pesky mouse dragons represent antigens that are found inside the cell. Once processed, these endogenous proteins are presented by MHC1 on the cell surface and provide a window into what is being produced inside the cell. As tasty as house mouse dragons are, they still require a bit of processing, and the first step is that. Newly produced endogenous proteins, again meaning proteins that come from inside the cell, are cut up into smaller peptides inside proteasomes. The fact that they're newly produced is helpful because it aids the T cell army in responding earlier to an infection. Now, placing that rather large cleaver aside, the cut up mouse dragon peptides are then poured out of their bowl, which requires some tapping to get every last bit out. This tapping represents the TAP transporter, which brings the peptides into the endoplasmic reticulum, or in this case, into an ER-shaped cooking mold. Finally, a special member of the kitchen team, wearing an MHC1 badge, serves the delicious meal to the killer knight waiting outside. This final step symbolizes peptides being loaded onto MHC1, moved out of the ER through the Golgi apparatus, and finally to the cell surface for presentation to CD8 positive T cells. Okay, let's see what's cooking up on level 2, where a package has arrived from afar, imported dragon meat. A delectable delicacy, to be sure. It's stamped with an imported label because MHC2 presents exogenous proteins, or those that are found outside the cell, to CD4 positive T helper cells. The big burlap sack holding the dragon meat is both industry standard packaging and sketchy symbol for the phagosome. The phagosome encases the exogenous proteins and brings them inside the cell. Now, simultaneously, you'll notice the 2-tasseled pillow, representing MHC2, getting moved along this Royal Kitchen's golden transport system by the invariant chain. The invariant chain has a few roles here. First, it helps the MHC2 move from the endoplasmic reticulum through the Golgi apparatus and to the endosome, and it also prevents interference from other unwelcome wandering peptides by blocking binding to MHC2. The large clip on top of the 2-tasseled pillow represents the clip portion of the invariant chain, which blocks the MHC2 binding site. Nothing else is allowed to rest on the fancy pillow. It's only for imported dragon meat. Meanwhile, the phagosome sac has spilled its imported dragon meat onto the cutting board, where the cleaver is chopping it up. This symbolizes the action of the phagosome combining with the endosome, the place where the exogenous proteins are chopped into bite-sized peptides. So tender. Supervising the finale of this process is the D-maître. Anyway, that's not mater D, it's D-mater, who's standing in here for HLA-DM. The DM has the honor of releasing the clip and allowing only the most tightly binding exogenous peptides to attach to MHC2. That is, they only allow the finest cuts of imported meat to be loaded onto the two-tassel pillow for service. And finally, the delicious pillow meat, or MHC2 peptide complex, is transported to the cell surface for presentation to the waiting and hungry helper squires, or CD4 positive T cells. Bravo, Castle Chefs. Bravo. It's truly a beautiful immunological and culinary process. But before we take a taste, let's do a quick review. MHC restriction refers to the fact that a T cell is only able to recognize antigen bound to a specific MHC molecule. CD8 positive killer T cells bind to MHC class 1, while CD4 positive helper T cells bind to MHC class 2. MHC1 presents endogenous proteins, those that originate from inside the cell. First, proteasomes cut up the proteins, then the TAP transporter moves the cleaved peptides into the ER. The peptides are then loaded onto MHC1 and displayed for CD8 positive cells on the cell surface. The MHC2 process is a little different. MHC2 presents exogenous proteins, or those found outside the cell. First, the proteins are enveloped in a phagosome and brought into the cell. Then, the invariant chain helps MHC2 move from the endoplasmic reticulum along the Golgi apparatus to the endosome. The clip, which is part of the invariant chain, temporarily fills the MHC2 groove to prevent other peptides from accidentally binding. The phagosome then combines with the endosome, and the proteins are chopped up into peptides. HLA-DM releases clip, allowing peptides to attach to MHC2. The MHC peptide complex is then transported to the surface of the cell for display to CD4 positive helper T cells. Dragon meat. Definitely an acquired taste. Would pair well with a full-bodied Syrah, I think.
Okay, so that is the difference between MHC1 and MHC2. Thanks to Sketchy for making the perfect video for me. I like their style. They get me. Anywho, MHC class 3 molecules include inflammation and the complement system. Guess what we're learning after the MHC? Yes, the complement system. And they are a group of proteins in the blood that help the body fight infection and damaged cells. Part of your immune system that defends your body against injury and foreign invaders like bacteria and viruses that can make you sick. Remember, the MHC class 3, they do not present antigens directly. They are triggered by inflammation signals inside the cells and from cytokines. The MHC class 3 encodes for some of the components in the complement system. The physical proximity suggests a functional link, potentially influencing the efficiency and regulation of both systems. So it could wind up influencing both the MHC3 and how it works with your immune system and your complement system with how it works with your immune system. These are the following basic functions of the complement system: opsonization, enhancing phagocytosis of antigens; chemotaxis, which is attracting macrophages and neutrophils; cell lysis, which means rupturing membranes of the foreign cells; and clumping, which are antigen-bearing agents. The major histocompatibility complex genes, also known as the HLA, are the prevailing contributors of genetic susceptibility to autoimmune diseases, type 1 diabetes, multiple sclerosis, rheumatoid arthritis, and it plays a significant role in the occurrence and outcome of infectious diseases such as malaria, parasite, HIV, the COVID. It can also cause epigenetic changes, the HLA-C, and there are plenty of others, trust me. If this system isn't working properly, it's going to have your immune system going haywire or not activating at all or misfiring with the wrong cells. So you want to make sure you take care of this.
In this lesson, we're going to talk about MHC molecules. There are two types, MHC1 and 2, and they both have the same goal: to present antigen fragments at the cell surface where T cells can see them and get into a murdery mood. Despite their shared goals, there are some important differences between the two, so we'll split the sketch in half. MHC1 will go on the left and MHC2 on the right. But first, let's talk about some commonalities that we'll symbolize here in the middle. MHC genes are located on chromosome 6, hence this 6 in the stone wall. And notice that the wall is made up of a whole bunch of different types of rocks. That's because MHC genes are super polymorphic, which helps contribute to an adaptable immune response. On chromosome 6, the specific genes that code for MHC are located in the human leukocyte antigen locus, or the HLA locus, represented here by the HLA banner touting Her Majesty's Loyal Army. Hooah! Or something like that.
So the HLAs are corresponding to MHC class 1 are HLA-A, HLA-B, and HLA-C. The HLA-A gene presents peptides for the cytotoxic T cells, or the killer T cells, or the CD8+ cells, which helps the immune system recognize and respond to foreign substances. HLA-A is involved in the immune response to infectious diseases like HIV, malaria, and SARS-CoV-2. The HLA-A is what doctors look at when they're considering doing a bone marrow or kidney transplant. The HLA-A has been associated with severe cutaneous adverse reactions and HLA modifiers. So what does this mean? HLA modifiers can impact the immune system, your cancer risks, especially breast with the BRCA1 and other health conditions. It can also impact how cancer antigens are presented to the immune system, which can impact cancer progression. The HLA B27 is associated with increased risk of developing autoimmune disorders and even some cancers. The HLA-B has many possible variants, which allows the immune system to react to a wide range of foreign invaders. They bind to peptides from the pathogens and cancers. They trigger the CD8 T cells. They trigger the infected cell to self-destruct. There are hundreds of versions of the HLA-B gene called alleles. Each allele is given a number, such as HLA-B27, etc., and get categorized together. One study found that HLA-B1501 is associated with asymptomatic SARS-CoV infection. Mutations in the HLA-B gene are associated with ankylosing spondylitis and schizophrenia. The HLA-C gene is involved with an immune response, including interactions with T cells and natural killer cells. It presents antigens to the T cells and binds to the receptors on the natural killer cells. The T cells, it helps the body recognize and respond to pathogens, and natural killer cells helps regulate their development and function. It's involved in many processes, including protection against viruses and cancer and the development of autoimmune diseases. Due to its central role in activating natural killer cells, HLA-C has been linked mainly to viral infections, autoimmune diseases, and cancers. The HLA-C can make epigenetic changes. It helps to establish tolerance to the fetus if you're pregnant. It is linked to many diseases including COVID-19 infection and severity. The HLA-C0401 is linked to a more severe course of COVID-19. It is also linked to rheumatic diseases, preeclampsia, organ and tissue rejection, myalgic encephalomyelitis, chronic fatigue syndrome. And the thing to understand here is, remember, the cycle of illness that we have been learning is behind all of these processes and how they work, that in your genetics. So balancing the cycle of illness is crucial to regaining control of these functions so that they work properly. It'll go a long way to helping you feel a lot better. The HLA-C602 genotype has been linked to the treatment response of psoriasis, and the HLA-C802 represents the KRAS G12 mutation, which is a common cancer mutation. It's time for How Is It All Connected? The major histocompatibility complex, class 1, works with mitochondria. Mitochondria DNA is subject to increased rates of mutation due to its proximity to the source of reactive oxygen species, that's oxidative stress, Mitochondria is subject to high mutation rates because of its proximity to the primary source of oxidative stress, or ROS. To prevent accumulation of more mitochondrial mutations, mechanisms take place that eliminate cells with mitochondrial defects. The MHC1 alerts the immune system to cells with mitochondrial mutations. It is overexpressed in fibroblasts with mitochondrial dysfunction from patients with mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes. Immune surveillance of DNA mutations in cases of immune failure occur as cancer, degenerative diseases, and aging. We are talking about premature aging and rapid aging due to illness. Methylation. Methylation also affects the MHC class 1. This can be directly affected by methylation. DNA methylation patterns can regulate their expression levels. High methylation can lead to decreased MHC class 1 gene expression, often observed in cancer. The downregulation of MHC molecules helps them evade immune detection. Hypermethylation, which means there are more methyl groups added compared to a normal state, which leads to changes in gene expression, often resulting in gene silencing, of the MHC class 1 can be associated with a decreased expression in various cancers. They are subject to epigenetic regulation, including DNA methylation. This can change their transcription levels without changing their underlying DNA sequence. When the regions that promote the MHC genes are heavily methylated, meaning too fast, so cut out those triggers, it can inhibit the gene expression. This in turn will further interfere with methylation and cause the cycle of illness to continue to spiral out of control. So it's going to also then create more mitochondrial issues. This can lead to types of cancers. This can wind up making your mast cells, chronic illness, and autoimmune disorders far worse. So please cut out your triggers and toxins. Because the major histocompatibility complex is the mechanism that winds up initiating immune responses, mitochondrial health and methylation are directly involved in the major histocompatibility complex. Your allergens, stress, lifestyle choices, diet, all directly impact this mechanism and your immune responses. So please change your habits and diet if you are suffering from immune issues. This plays a huge part in your symptoms and condition. This is just the tip of the iceberg, but you can see that they're involved with one another. The gut microbiome also significantly influences the MHC complex 1, or class 1. Microbiome shapes the expression and function of the MHC molecules on immune cells within the gut lining. The microbial dysbiosis plays a big role in how the immune system reacts, which leads to susceptibility to diseases. Diverse bacterial species present in the gut can stimulate or suppress immune cell activation depending on their composition. The microbiome directly interacts with dendritic cells, which expresses MHC molecules. This affects their maturation and antigen-presenting capabilities, i.e., your microbiome has a direct role in how your immune system works. Different bacteria species within the gut microbiome produce different antigens to the immune system. These are displayed on the MHC1. This mechanism shapes the way the immune system responds to specific bacterial components. A balanced microbiome helps maintain immune tolerance by promoting the development of regulatory T cells, which can prevent excessive immune responses against the commensal bacteria. In inflammatory bowel disease, dysbiosis or imbalance in the gut microbiome can lead to altered MHC expression. So that means if you're not taking care of your gut, you can wind up changing the MHC and how it works inside of you and make all of your symptoms a whole lot worse. This is often why when people start eating properly and heal their gut, start feeling better. It can also contribute to the development of inflammatory conditions like Crohn's and ulcerative colitis. So, aberrant immune responses triggered by the imbalanced gut microbiota can trigger the development of autoimmune diseases by influencing the health and presentation of self-antigens. The gut microbiome can also influence the development of allergies. by shaping the immune response through the MHC interactions. So you need to keep a healthy gut microbiome because it's directly going to change how your MHC works. And this is going to have a drastic effect on your health. That's why a lot of people, when they heal their gut, feel a lot better. Because you altered the MHC functions when you healed your gut. Depending on the specific MHC alleles an individual possesses, certain bacteria species may be more readily targeted by the immune system, leading to a unique microbiome composition. Look at it with the case of IBS and allergies. Our immune system are attacking specific triggers. This in turn changes our microbiome and how it works with the immune system. It also goes the other way. The MHC, because you don't have a well-diverse microbiome, it changes the way the MHC works in your body, and that can create more allergies in this example. This can wind up contributing to why people can't, why folks have such a trouble tolerating foods, or they get to the point where they can almost eat nothing. Healing the gut can fix some of these problems and help you feeling better. Certain MHC variants may be associated with increased susceptibility to specific diseases as well. Studies have shown that folks with different MHC genotypes have distinct gut microbiota compositions, which can be linked to immune response and disease risk. Research in animal models have demonstrated how mutations of MHC genes can directly influence the development and composition of the microbiome. So, of all of this, what do you need to know? You get your microbiome and mitochondria from your mother. If you keep the bad habits, triggering foods, bad diet, no exercise, extreme stress, this can all change how your microbiome works and how your immune system responds through the MHC complex. You must take care of your health, or it'll lead to autoimmunity through these processes. So remember, this ties with mitochondria and methylation. I've said it again and again. Fasting and rebuilding the microbiome from scratch can help solve a lot of these problems, but it takes a lot of time, work, and effort, and a lot of patience and unwavering discipline and determination. I'm not gonna lie, it's not easy. I did create healing plans to achieve this. It's free, and you can find it on my websites. It's the healing checklist. Just make sure you read everything before you start it. The MHC class 2 is found primarily on antigen presenting cells, or APCs, and those are like your B cells, macrophages, and dendritic cells. B cells, the MHC2 plays a crucial role in activating the B cells. It presents antigens to the helper T cells. So the B cells present antigens to the T cells. The MHC2 on B cells acts as a signal to the T cell to initiate the activation process of the B cell itself. So it's kind of like the B cell goes to the T cell and says, hey, we need to activate, and then the T cell says, yes, we ride or die, and then they go together, basically. And then the B cell gets all excited because now it's got a partner to go, and it activates itself and gets to work. They hype each other up. B cells express MHC2 molecules on their surfaces to present processed antigens to the CD4 plus T cells. Remember, the B cells are triggering the T helper cells. And when a T cell recognizes the antigen on the MHC2 complex on a B cell, it releases cytokines that stimulates the B cell to activate and proliferate. The MHC2 can also directly signal within the B cell. So the MHC2 directly contributes to the B cell to be able to activate and differentiate to what it needs to be. The MHC class 2 also presents antigens to the CD4 plus lymphocytes. Those are your T helper cells. So this helps activate the T cells and differentiates them into T helper cells. The T helper cells can only bind to MHC class 2. They wait to collect their antigen, and they are found outside of the cells and brought to the cells to be presented in the CD4 plus T cells. So the T helper cells can only bind to the MHC class II. Like we learned in the video we watched a few minutes ago. They captured the dragon mice outside the castle and imported them in, or outside the cell. This video is describing what we're talking about now, and is a perfect representation. So, the MHC class II helps control autoimmune responses in humans. The HLAs corresponding to the MHC class II are the HLA-DR, the DP, and the DQ. MHC class II molecules also activate intercellular signaling pathways, frequently leading to apoptosis, which is cell death. So if there's something wrong with the cell, it can do it itself. Expression of the MHC class II molecules is confined to the professional antigen-presenting cells, APC, of the immune system, and in non-professional APCs, MHC class II molecules can be induced by a variety of immune regulators. A professional antigen-presenting cell is a specialized immune cell, like a dendritic cell or macrophage, that is highly efficient at presenting antigens to T cells and actively initiating immune responses. A non-professional APC is a cell that can present antigens but does so less effectively and only under certain conditions, like epithelial cells or endothelial cells, and typically lacks the necessary co-stimulatory molecules to fully activate T cells. The key difference lies in their level of the MHC class II expression and ability to robustly stimulate T cell activation. So the non-professional APC is like a weaker version of the professional one. So if you think about at home, if you're thinking about hiring a professional to do something in your house versus trying to do it yourself, that's the difference between professional antigen or the non-professional antigen. The professional is more efficient, can get it done faster, better, etc. Whereas the non-professional is the jack of all trades and it's trying to help, but it might not actually be able to do it. And the key defining factor is how much MHC class II they are expressing. It's that time again for How Is It All Connected? So the MHC class II plays a part in mitochondria. Mitochondria indirectly influence the antigen processing pathway. Mitophagy is a degradation of damaged mitochondria, where the mitochondrial proteins are released and processed into peptides. Mitochondrial function is tightly linked to cellular metabolism, which is critical for the activation and function of immune cells. Proper mitochondrial activity is necessary for the APCs to efficiently process and present antigens on the MHC class II. Mitochondria contributes to the generation of a pool of peptides that are ultimately presented on the MHC class II to activate helper T cells. The movement and interactions of mitochondria within a cell can influence how efficiently antigens are delivered to the MHC2 loading compartment. It has a metabolic function. The production of ATP, that's what makes your energy, is crucial for the functioning and antigen-producing cells. They also include their ability to efficiently process and present antigens on MHC class II molecules. Mitochondria plays a significant role in the regulation of innate and adaptive immunity, so the health and activity of mitochondria can significantly impact the process of antigen presentation on MHC2 molecules. The antigens on macrophages and dendritic cells are critical for initiating the adaptive immune system, so damaged mitochondria can lead to changes in the protein processing and availability of peptides to be displayed on the MHC2. Remember, mitochondria makes those peptides, so damage is going to change how your immune system responds. And speaking of peptides, all those peptides on television, you're taking them. You're not fixing the root problem. You're just adding a band-aid. When mitochondria are under stress or dysfunctional, they can release damage-associated molecular patterns, or DAMPs, which can trigger the immune system by signaling APCs to increase the MHC2 expression. This is why it's crucial to take care of your mitochondria and avoid allergens. It creates an inflammation and an immune system response that can get out of control, and then it ramps up your MHC2 expression, which means more immune responses and aberrant immune responses. So people with chronic illness and especially mast cell disorders, so your cells are sending out the danger signal, and that's because of your damage to the mitochondria. And then it ramps up the MHC2, which then winds up calling more cells. This is why fasting is important, to clear out the damaged cells to help reset the body. It stops all these aberrant processes. Aberrant mitochondrial function can contribute to the development of autoimmune diseases by causing the immune system to recognize self-antigens from the mitochondria as foreign, which leads to the autoimmune response against healthy cells. So, conditions that disrupt mitochondrial function can lead to altered antigen presentation on the MHC2. This will affect the immune tolerance. So if you're a mast cell patient, this process will just spiral out of control. It contributes to autoimmune diseases. Stress and or damage can help cause this. So just being under chronic stress and serious chronic stress can make it 1,000 times worse. This can all lead to further illness and intolerances. So your mitochondria and stress and the MHC complex, man, they go together really tightly. So to get out of the danger zone and you have a mast cell disorder, fasting is the best and fastest way to do this. I know it sucks, but you want to fix your health. Live with the pain of trying to heal now or the pain of disease later. The MHC2 affects methylation. It typically downregulates the expression of the MHC class II by silencing the class II activator or cytogene through DNA methylation. The cytogene is a gene within the major histocompatibility complex that encodes a protein which acts as a master regulator for the transcription of the MHC class II genes. This controls the activity of the MHC2 genes by coordinating various proteins to turn on. This in turn affects how your immune system responds. It helps other proteins to activate gene expression without directly binding to DNA. That's a good thing. Mutations in the cytogene can result in Barr lymphocyte syndrome type 2. The immune system cannot properly produce a class of proteins, leaving the body vulnerable to infections. Now, please don't get this confused with the Guillain-Barré syndrome, because that's a rare neurological disorder where the body's immune system attacks its own nervous system. And this attack can damage the myelin sheath protecting and around the nerves, leading to muscle weakness, tingling, and sometimes paralysis. And Guillain-Barré syndrome is an autoimmune condition. But everything is connected, so this would lead to that worsening indirectly. This effectively prevents the MHC2 genes and reduces the presentation of the antigens to immune cells. DNA methylation of the CPT location within the CITA acts as a repressive signal, inhibiting the CITA gene, which is crucial for the MHC class II expression. So what does this mean? If we don't take care of our bodies, heal methylation, mitochondria, and our gut health, which also plays a part, we're going to change the way our cells tell the immune system it needs to help or protect us. The cavalry, aka the immune system, won't come to save the day if this is a problem. The MHC and the microbiome also are connected as well. Gut health significantly impacts MHC2 expression on intestinal epithelial cells. This allows these cells to present antigens from the gut microbiota to the T helper cells, which plays a crucial role in maintaining immune tolerance and regulation of inflammatory responses within the gut. A disrupted gut microbiome can lead to altered MHC2 class expression, potentially contributing to the development of inflammatory bowel disease and other immune dysregulations. Intestinal epithelial cells, which form the barrier between the gut lumen and the immune system, express MHC2 molecules, enabling them to present antigens from the gut microbiota to immune cells. So, if you have IBS or any other type of gut where you have significant inflammation in the gut, your MHC2 is involved, which also means you have a weak or dysregulated microbiome. So the composition and diversity of your gut microbiome directly influences the level of MHC2 expression in the intestinal epithelial cells. So if you don't have a proper microbiome, you are going to alter the MHC class 2, and this can result in you being more allergic to things, being more responsive to things, and having way more sensitivities, or even attacking your own microbiome. If you have IBD, IBS, any of the above, you need to cut out your triggers, don't smoke, and you have got to reduce your stress. You would be surprised how much stress messes with this. Proper MHC2 expression in intestinal epithelial cells is crucial for maintaining immune tolerance towards commensal bacteria, preventing excessive immune responses against harmless microbes. In conditions like IBD, where the gut inflammation is present, MHC2 expression on the intestinal endothelial cells can be dysregulated, leading to abnormal immune responses against gut bacteria. Cytokines produced by immune cells in the gut can also influence MHC2 expression on intestinal epithelial cells, further modulating immune responses. Again, this is why so many people wind up improving when they fast, and then we rebuild the microbiome so that we can stop all of these aberrant functions. MHC class 3. As with all MHCs, they are located on chromosome 6, which encodes proteins primarily involved in regulating immune response functions. They work with the complement system, which is C2 and C4. According to the NIH, it does C3, C4, and C5 as well. And basically what it does is the MHC contains genes that code for those components of the complement system, among other immune-related molecules. The complement components then contribute to the activation of a complement cascade, playing a role in inflammation and pathogen clearance. They also work with the inflammatory cytokines, tumor necrosis factor, and involved with heat shock proteins. Unlike MHC1 and 2, who present to T cells, MHC3 do not present antigens, nor are they involved in antigen production. They are only triggered by inflammation signals inside the cells and cytokines. They do play a big role in inflammation, though. They are upregulated during infection, foreign invasion, and cell damage. It is the only MHC that has the genes that codes for the complement system. This has a major role in triggering mast cells. C3 and C5 of the complement system triggers the mast cells. If cell lysis isn't working properly, you have chronic mast cell degranulation. And if MHC1 and 2 are not working properly, neither will the MHC3. This leads to cascading immune responses and chronic mast cell degranulation. It is time for How Is It All Connected? Methylation significantly affects the expression of the genes within the MHC3 by acting as a regulatory mechanism. It impacts the production of the complement system proteins that affects the immune response. Overmethylation in the MHC3 regions can suppress immune response activity. MHC3 region contains genes like the tumor necrosis factor, which plays a crucial role in inflammation and immune response. Aberrant methylation is a pattern of DNA changes that can lead to cancer and other diseases. You can have hypomethylation, which is undermethylation. In mast cell patients, this is generally you. You can have hypermethylation, which is gene-specific overmethylation. Some of you understand this as fast or slow genes. And some have known these aspects to be called dirty genes. Environmental causes, exposures, aging, diet, stress, poor sleep, and genetic variations in methylation will affect this. And I made a whole video on methylation and broke down each cycle and how they all work together along with the genes. There are also methylation slides and genes and SNPs slides. Aberrant methylation and DNA changes will cause cell cycle control, including mitochondria problems, altered transcription of factor functions, altered receptor function, disrupted cell-to-cell interactions, and inactivate the tumor suppressor genes. So it's really important that you keep your methylation under control because it's going to have a direct link to your MHC class 3, which is constantly going to be also affecting your complement system. And your complement system is like the global cleanup team, and it kicks in as a backup to your immune system. Mast cell patients, this is most definitely happening to you. It works in all illness, but for mast cell patients, it keeps you stuck in chronic degranulation. So this can put you into what is called cell danger response. It is the cell's defense mechanism and can be triggered by threats like infections, toxins, physical trauma, or psychological stress. For healthy individuals, this is a normal process when the cells are damaged and the body and cells try to fix it. Generally, you're none the wiser. However, if you have chronic illness and you don't take out your triggers or stop the chronic stress, the cells don't get to finish healing, and it will contribute to significant chronic illnesses and conditions, as well as chronic mast cell degranulation. This will also put your mitochondria at serious risk. The major histocompatibility complex class 3 and the complement system, don't worry, we're going to be talking about the complement system in a minute, but the MHC class 3 and the complement system play a major role in the cell danger response and the associated inflammatory and immune responses. But these processes can seriously damage mitochondria and will impact the cycle of illness negatively and further drive it out of balance, which then in turn further alters your major histocompatibility complex and your complement system. And don't forget how much this is going to impact your microbiome and how much your microbiome in turn affects it. So are you starting to understand why I needed to teach this? Because this is also going to greatly affect your cells in your immune system. And this includes your T cells, which are going to go on little killing sprees and attacking you instead of the foreign object. Because you can either be healing or you can be fighting. You cannot do both at the same time. And that's why it is so important to make sure you get your body to homeostasis or a complete rest. So if a cell has too much damage, then it produces antigens. And this will in turn activate the killer T cells, which regulates the cell death, which can produce more DAMPs or damage-associated molecular patterns. If you don't allow your body time to heal and eliminate your triggers and stress, this will cause serious symptoms and cause disease or cancers. Cell damage response 1 is going to include allergies, asthma, and atopy. And atopy is a genetic predisposition that makes individuals more susceptible to allergic reactions, particularly to the common environmental allergens such as pollen, dust mites, pet dander, and mold. And conditions related to atopy are eczema, asthma, and allergic rhinitis or hay fever. Cell danger response 1 will also cause chronic infections, which include fungal, bacteria, viral, and parasitic. Cell damage response 2 results in metabolic syndrome, inflammatory bowel disease, benign prostatic hyperplasia, cancer, and leukemia. And for metabolic syndrome, that also means it royally messes with your methylation cycles. And for cell damage response number 3, you're going to have symptoms like chronic fatigue syndrome, fibromyalgia, chronic pain syndromes, generalized anxiety disorder, major depressive disorders, post-traumatic stress disorders, autism spectrum disorders, migraine headaches, POTS, Parkinson's, Alzheimer's, autoimmunity, amyotrophic lateral sclerosis, osteoarthritis, and irritable bowel syndrome. If you can't eat any foods, even after trying to heal, This is why you are stuck in the cell danger response. The only way I know how to get out of this successfully and sooner rather than later is to fast, because your body can't fight and heal at the same time. By fasting and drinking water, you are giving your body a break and allowing it to catch up. But you can't start eating again too soon, or it's going to keep your cell danger response going. And this applies to all of your triggers. So especially mast cell patients, we tend to be more knowledgeable about what our triggers are. So you need to make sure that you cut out all the triggers that are causing immune responses, or in your case, mast cell flares. Just because you stop eating, if you're still getting the onslaught of other triggers and you have chronic stress on top of that, you're not going to be able to heal the cell danger response. So you need to fast and make sure you get your stress under control and get rid of all of your triggers. If you are finding yourself with some serious neurological pain, fatigue, chills, motor function issues, cognitive function issues to an extreme, and any food will cause worsening neurological issues, or you just can't eat anything anymore, you are in the danger zone of the cell danger. You must take this seriously or it'll cause worse condition. I am telling you this because I don't want you to make the same mistakes I did. And for severe symptoms, I am talking about temporary blindness, temporary paralysis, pseudo-stroke-like symptoms, Alzheimer's dementia type symptoms, and or you're getting MS or Parkinson's type symptoms. You need to take this extremely seriously and get out of the danger zone ASAP. Especially if you're getting the severe brain and spine burning and pain. So as we were discussing, the major histocompatibility complex and methylation go hand in hand. The associated diseases when these processes go wrong are cancer, breast, lung, renal, cell, skin, and neuroblastomas, developmental defects, obesity, asthma, and neurodegenerative diseases. Aberrant methylation can be hereditary and passed down through the generations. Look at your methylation and your detox profiles. The MTHFR gene mutations and associated SNPs in methylation are becoming more and more popular in the medical industry, although a long way from application unless you do it yourself. I explain all of this in the methylation video and slides. And remember, you don't have to have these mutations in order for them to not be working properly. All it takes is these cycles getting out of balance to cause a whole cascade effect. Those with the MTHFR just have to take extra steps to get healthy. And those that don't have the MTHFR mutations, oftentimes it's because you're stuck in cell danger. Aberrant methylation can actually predict future lack of gene expression. That's why it's important to cut your triggers now and prevent any further epigenetic changes and shortfalls for the future. This has a long-lasting, long-term effect if it's not fixed. The major histocompatibility and the microbiome have a relationship. Now, it's not directly involved with the major histocompatibility class 3. However, it dictates how the immune system works. And if the MHC1 and 2 are not working, which it does affect, MHC3 will not work properly either. The MHC class 3 also has mitochondrial involvement. Dysfunction of the mitochondria can lead to altered MHC expression due to the disruptions in cellular metabolism and the reactive oxygen species, which means you're going to have more oxidative stress. Proper mitochondrial function is necessary to generate ATP, which is your body's energy. ATP is needed for cellular processes involved in antigen processing and the transport of the MHC molecules to the cell's surface. If ATP production is low, it decreases antigen processing and presentation, and a lower MHC expression on cell surfaces. The changes in the metabolic pathways like glycolysis or oxidative phosphorylation can impact the MHC. You can see the picture in the top right-hand corner, and you can see the mitochondria and the Krebs cycle and the oxidative phosphorylation that's on the right side. And you can see how the mitochondria directly feeds into the methylation cycles. So the methylation cycles and the mitochondria play a direct role in how your major histocompatibility works. Cancer cells often exhibit altered mitochondrial function, which can be used as a mechanism to down-regulate MHC expression and evade immune detection. This is why it's important to cut out your triggers now and prevent any further epigenetic changes and shortfalls for the future. Control the triggers you do have control over. T cells are also under the mercy of mitochondrial health. Okay, the complement system. As promised, I told you I would talk about it. And bear with me, stay with me, you'll understand why this is so important. The complement system also works with the MHC class 3. That's our major histocompatibility complex. Alright, so the complement system is a network of proteins that helps the body fight infections and diseases and helps it heal. It is a part of the innate immune system. It also helps shape the adaptive immune response. It's like a backup team in your immune system. It complements the work of other immune cells to fight off infections effectively. The complement system is part of your body's immune system that cleans up damaged cells, microbes, and immune complexes, helps your body heal after injury or an infection, destroys microscopic organisms like bacteria that make you sick. It promotes inflammation and attacks the cell membrane of pathogens. The complement proteins float around the blood in an inactive state. When activated, they change shape to get to work in a chain reaction that activates the next protein in line to turn on, just like how one domino falls into another. This affects how the immune system works to help the body fight and heal. This chain reaction is called a molecular domino effect. This chain reaction leads to immune system activation and amplifies the response. A number of complement proteins are proteases. Those are enzymes that breaks down the proteins and peptides that are themselves activated by proteolytic cleavage. And that is a process of breaking the peptide bonds between amino acids and proteins. Don't worry if you don't understand that. It'll make more sense later. The complement system can be activated through 3 pathways. The classical, which is triggered by antibodies bound to a pathogen. The lectin pathway, which is triggered by sugar molecules on pathogens, or PAMP recognized by lectins. And remember, PAMP means pathogen-associated molecular pattern. And the alternative pathway, which is activated spontaneously when encountering a foreign surface. So the complement system can be activated through 3 main pathways: the classical pathway, the lectin pathway, and the alternative pathway. Think of the complement system like a home security system with different ways to trigger the alarm. The classical pathway, the alarm triggered by antibodies, this pathway will get activated when antibodies, your immune tags, attach to a target, like bacteria. It's like your security system goes off because it recognized a tagged intruder. With the classical pathway, the antibodies must already be there, such as the IgG or IgM, meaning this is the second time response or adaptive immunity kicking in. The lectin pathway is the sugar sensor. It functions similar to the classical pathway but gets activated when some bacteria have sugars on their surface. Your immune system has a lectin sensor that recognizes these sugars and sets off the alarm. It's like your system recognizes a red flag pattern on the intruder's clothing, or a sugar coat. For the lectin pathway, no pathways are needed, just detection of the sugar patterns. So these do not require antibodies to trigger them. The alternative pathway, or the default patrol, this one's like a constant surveillance. It randomly checks surfaces, and if it doesn't find a self signal on something, it attacks. So it's like your system is saying, this doesn't belong. Attack first, ask questions later. This works all the time in the background. Quick, dirty, and innate. So the classical pathway requires antigens, like IgM or IgG. The lectin pathway looks for a sugar-coated pathogen, and it responds then. and the alternative pathogen constantly runs patrol, looking for anything that does not identify as self. Leaky gut can make that worse. So, the major functions of the complement system is opsonization, inflammation, cell lysis, and clearance of immune complexes. So let's take a brief look at each one of them. Opsonization marks pathogens for death. The C3b coats pathogens, making them easier for phagocytes, which are macrophages and neutrophils, to engulf. We've seen this video a couple times now, and again the pathogen gets engulfed in the phagosome, and the bacteria or pathogen winds up getting destroyed. One of the functions of the complement system is inflammation. It attracts macrophages, neutrophils, and mast cells. The C3a and C5a act as anaphylaxitoxins, recruiting the immune cells. They increase blood vessel permeability and creates inflammation. The complement system also does cell lysis. It is the membrane attack complex, or as I like to call it, the MAC attack. This includes the C5b, C6, C7, C8, and multiple C9 proteins. Do not worry, we will be discussing what these are in a few minutes, but it's important that you know what these pathways are. These form pores in microbial membranes, causing cell death. It acts like a special ops military team going in and fighting the foreign cells by making the membranes of cells rupture, thus destroying the microbes.
Complex is part of the complement system, which exists to aid in inflammation, phagocytosis, and other aspects of the immune defense, including pathogens from the body. There are two primary stages in which the complex is formed. The first is referred to as initiation. During initiation, protein C5b binds C6 and C7. The newly formed C5b67 complex binds to the membrane by C7. The second stage, through which the complex destroys a pathogen, is referred to as the polymerization stage. C8 binds to the complex and is able to insert itself into the membrane because of traits relating to the polarity of the molecules that make up the membrane and the protein itself. C8 is then able to induce many C9 proteins to form into a porous structure that penetrates the cell surface, filling it with water and oils, inevitably destroying the cell.
And the complement system is also in charge of clearance of the immune complexes. This complement helps remove antigen-antibody complexes, which prevents an autoimmune response. The complement system is a powerful immune mechanism that enhances both innate and adaptive immunity by marking pathogens, recruiting immune cells, and directly lysing infected cells. Its regulation is essential to prevent excessive tissue damage and autoimmune diseases. If you don't clear out these immune complexes, you're going to wind up having hypersensitivity issues and autoimmune conditions because you're leaving all these markers behind telling the immune system to keep going, and what happens is it's not getting cleared out to allow the immune system to shut down, or at least stop attacking. The complement system is made up of over 30 proteins. These are produced primarily by the liver, so you can imagine if you have an unhealthy liver what's going to wind up happening. They present in the blood and tissues in an active form. When activated, they trigger a chain reaction that amplifies the immune system. They are a front line of defense. Think of them like special ops teams that get together to work together and defeat foreign invaders, pathogens, and bacteria and microbes. So in this video we watched earlier, these are a bunch of proteins coming together, and these are different types of proteins coming together to work together. And then what winds up happening is as they work together, the ultimate goal is to penetrate that cell and to fill it or lyse it with oils and water to destroy the pathogen. The key components include the complement proteins 1 through 9. This is C1 through 9. These proteins form the core of the complement system and participate in sequential activation process. Examples: the C3, the central protein that gets cleaved into C3a, which is an inflammatory response, and C3b, which is opsonization, and C5 cleaved into C5a, inflammatory, and C5b, part of the membrane and attack complex. There are complement receptors that are found on immune cells. These receptors recognize complement proteins and help mediate immune responses. Examples are the complement receptor 1, 2, 3, and 4, which are found on macrophages, neutrophils, and B cells. And there are complement receptor on C3, C5, which are found on mast cells. The complement complex consists of a series of proteins, C1 through C9, that work in a cascade-like fashion to eliminate pathogens through opsonization, inflammation, and direct lysis. So like we said earlier, when activated, they trigger a chain reaction that amplifies the immune response like dominoes falling into place. Now, I don't want to get in depth with this because I'm afraid of confusing you. So I'm just going to hit the highlights on each one, but you can pause the video and read through this, or you can find it in the immune system slides. So C1 basically recognizes and binds to antigens or antibodies that are having the IgG or IgM. This is part of the classical pathway. The C2 through 3, 4 create inflammation. The C3 is a central protein in the complement cascade. C3a acts as an anaphylaxitoxin, promoting inflammation and recruiting of immune cells. C3b is essential for opsonization, marking pathogens for destruction, phagocytosis, and forming C5 convertase. C5 also releases strong anaphylaxitoxins. It also recruits immune cells. Mast cells come into play with the C3 and C5. Mast cells. And they also initiate the MAC attack or MAC formation. That's when all those proteins were coming together to drill into the cell. And C6, C7, C8, and C9 are the formation of the membrane attack complex, and they're like a military infiltration team, like SEAL Team 6. These folks hit their target, penetrate, and annihilate the foreign microbes. So C6 stabilizes the whole MAC attack assembly. So all those towers you saw in those rings, it stabilizes all those formations. C7 inserts into the microbial membrane, so it attaches to the cell. C8 further embeds into the membrane and starts pore formation, so it's starting to drill in. C9 forms a pore-like structure in the microbial membrane, leading to cell lysis and death. So the MAC attack is C5b, C6, 7, 8, and multiple C9 proteins. If you want to keep your complement system happy, avoid allergens, trigger foods, maintain a healthy microbiome, take care of your diet, your stressors, your allergens, etc., and exercising, you can keep your gut healthy, and therefore the cycle that governs all illness will work properly, thus keeping your immune system in check and you at the happiest and healthiest you can be. Avoid sugar and other gut-busting ingredients like potatoes, guar gum, xanthan gum, smoking, drinking, etc. These will have a huge, huge impact on your complement system. Okay, why did I just teach you about the major histocompatibility complex and complement system? Because we need to understand where we've been and where we're going. We can't proceed without you having a basic understanding of it, but most importantly, how it's all going to tie together. There's going to be a lot of aha moments coming up here in a second. Now, I've tried to keep these two concepts, though, as simple as I possibly can, so bear with me. It's that time again for How Is It All Connected? Alright, methylation. This affects genetic expression, right? And can make epigenetic changes and metabolic changes for self-preservation. So if you're having a rough time with your health, your body is already making changes to help protect you. It affects processes in the body, and it affects the ability to detox efficiently. Methylation also turns on the MHC complex 1 and 2 and regulates it, and it's triggered by antigens. This also can make epigenetic changes. So you got methylation that can make epigenetic changes, and you got that major histocompatibility complex that can change or make epigenetic changes. So all methylation happens in the liver, and this affects your cell health, your microbiome,
and affects your gut health homeostasis.
And diet will alter this, stress, exercise, especially if it's too much or not enough, and the inability to maintain homeostasis due to a hampered pancreas, spleen, or gallbladder can mess this system up. So if you know you're having gallbladder, spleen, or pancreas pain, you're messing with your methylation and you're messing with your major histocompatibility complex, both of which could help control your immune system. The microbiome, it affects neurotransmitters, hormones, vitamin production in the microbiome, which feeds cells and helps methylation processes. The more damaged the gut, the more damaged the processes are. The diversity of the microbiome and its products it produces greatly affects cell health, immune function, detox function, and full circle back to methylation. It affects the immune system maturation, changing the way your immune system responds. It also affects the major histocompatibility class 3 expression and how often it gets activated. The microbiome can indirectly affect the class 3 by influencing the immune environment rather than directly altering the genes themselves. Dysbiosis can disrupt normal regulation of major histocompatibility class 3 gene, contributing to more inflammatory diseases. Dysbiosis can also change the response to pathogens. This is very important to understand. Your gut health is directing all of this, and it all ties into the cycle of illness. Your immune system activates the MHC class 1, which activates T cells, and the class 2, which are the B cells. Methylation, mitochondria, and gut health play a major role in how these processes work. Major histocompatibility complex 1 and 3 calls for more mast cells, which means higher histamine. Let me repeat that for the kids in the class. MHC 1 and 3 calls for more mast cells. Methylation governs it, microbiome governs it, the complement system and the major histocompatibility complex all go together. All lymphocytes produce histamine. Those are your B cells, T cells, mast cells, macrophages, and dendritic cells. They release pro-inflammatory cytokines and inflammatory signals within cells that trigger the MHC class 3. Did you get that? They release cytokines and inflammatory signals within the cells that trigger the major histocompatibility class 3. So let's look at this diagram. You have methylation. It has somewhat direct link in your gut health via the liver, gallbladder, and pancreas. Poor detox of the intestinal tract, gallbladder, affects your microbiome. Your gut health microbiome has a direct impact on your immune system and how it works. Your immune system releases cytokines and inflammatory signals within the cells, which triggers the major histocompatibility class 3. The major histocompatibility class 3, activated by cytokines and inflammatory signals and upregulating during infection, foreign invader, cell damage, and triggers the complement system. Because the complement system is a domino effect, C3 and C5, when it gets to that stage, triggers mast cells and impaired cell lysis. Now remember, cell lysis is that team that goes to the cell and drills into it and does the MAC attack and gets rid of the pathogen or the damaged cell or infected cell. So when the complement system triggers the C3, C5, we're triggering mast cells and impaired cell lysis, which means it's harder for you to fight infections. Then mast cell activation is triggered, and if cell lysising is not working properly in the complement system, then you have uncontrolled mast cell degranulation. So like everything else I've been teaching you, everything is a cycle, and it goes around and around, and it can either break or make your health. But you can see here how this can get you into a vicious cycle. This also keeps you in the cell danger response. Okay, the MHC class 3 in here, it is activated by the cytokines and inflammatory signals within a cell. That's the tumor necrosis factor alpha and the interferon gamma. And they are key regulators of the major histocompatibility class 3. When the immune system detects a pathogen or inflammatory stimulus, genes within the MHC3 region are upregulated to produce necessary complement proteins. So it's working with your complement system, and your complement system is coming in to help. This leads to the increased production of proteins encoded by the class 3 genes, particularly components of the complement system like C2 and 4, factor B, and cytokines like tumor necrosis factor and interferons, which this is essentially activating the immune response when a pathogen is detected. And the MHC class 3 is upregulated during an infection. It plays a major role in eliminating pathogens by facilitating their utter destruction. If MHC 1 and 2 aren't working properly, MHC 3 isn't going to work properly either. Methylation, mitochondria, and gut health directly affect how MHC 1 and 2 work. And if not working properly, it will have more inflammation signals and cytokines, etc. in the body, thus creating more immune signaling. Which means if there are more cytokines and inflammation, either through poor diet, infections, dysbiosis, stress, and allergens, smoking or drinking, etc., you'll have more activation of the MHC3 complex, which includes making more mast cells, which means it's further degrading the whole entire cycle of illness. And that's when the complement system comes in for the optimization, the inflammation, and remember, the inflammation attracts macrophages, neutrophils, and mast cells. And then the C3, C5 act as anaphylaxatoxins. I want you to remember that word, anaphylaxatoxins, because this can build up in your system, and that bucket we're always talking about, when our bucket gets too full, then we have a full immune reaction or mast cell degranulation. Well, if you get too many toxins built up in your system, guess what's going to happen? You're getting yourself deeper and deeper into a vicious cycle that's going to be harder to climb out of. Then you've got cell lysis. It also has to do cell lysis, and that's the one, remember, it's the membrane attack complex, or the MAC attack. And the MAC attack forms pores by drilling into microbial membranes, causing cell death. And it acts like a special ops military team going in and fighting the foreign damaged cells by making the membranes of the cell rupture, thus destroying the microbes. And then you have the clearance of immune complexes. This is vital to keeping you healthy because it helps prevent further mast cell degranulation, hypersensitivity reactions, chemical sensitivities, as well as autoimmune conditions. If this cycle becomes a runaway freight train, this is going to keep you also stuck in cell danger response. And that is very important that you get out of it. Now, to stop all of this, you basically have to fast and give your body time to clean itself up, fight any pathogens or foreign substances that are still in your body, remove all the antigens, and clearing out any more immune complexes left behind. This affects everyone. It doesn't matter what your health condition is. But mast cell patients, you want to pay attention to this. This is where the great cycle, I can't talk today. Alright, let me try that again. This is where the vicious cycle comes in. More and more mast cell degranulation, furthering the degradation of methylation, mitochondria, and gut health. And eating triggering foods or too much junk food is not going to help you at all, thus furthering the dysfunction in the MHC and the complement system. Now, for those people who are always writing in and telling me that they can only eat potato chips or drink soda, and that's the only thing they can tolerate. And the reason behind that is because you have the microbiome specifically to tolerate that and nothing else. That's why. So we have to fix that, because if you keep eating your potato chips and your soda, you're just going to continue to make this cycle worse. So I need you to understand that that is telling you right then and there that you have such a horrible dysbiosis in your gut that it can only handle your chips and soda, because that is what you have trained your gut to do. So now we have to fix it. And like I was saying, if cell lysis in the complement system is not working properly, it can lead to further mast cell degranulation, because the complement system produces molecules like the C3A and the C5, which are potent activators of mast cells. Even if the cell lysis function is impaired, essentially, the inflammatory response triggered by the complement cascade can still occur without the full cell killing effects, leading to increased mast cell activation and histamine release. The complement system, when activated, generates various fragments, including the C3A and C5, known as anaphylaxitoxins. Now, anaphylaxitoxins have the ability to directly stimulate mast cells to degranulate and release histamine and other inflammatory mediators. With cell lysis, the terminal pathway of the complement system forms a membrane attack complex that creates pores in the cell membranes, leading to cell lysis. Remember, cell lysis is where we are drilling into that cell and dumping in oil and water to make it rupture.
Okay, so if cell lysis function is compromised due to a deficiency in a complement component or regulatory issue, so basically it is saying if you have any problems in this area that we've been discussing. So if cell lysis isn't quite working properly, a deficiency in certain complement compounds or regulatory issues, which can be regulated by this system here, the earlier stages of the complement cascade can still happen. So just because we can't drill into the cell doesn't mean that all the forces don't come to the cell to try to kill it. If that happens, it's going to result in the production of more C3 alpha and C5 alpha. Those are your complement proteins that are helping you do all this. And this can still trigger mast cell degranulation, leading to an exaggerated inflammatory response. And exaggerated inflammatory responses and cytokine release continues to activate and aggravate the MHC class 3, which in turn continues to trigger the complement system, and which in turn continues with mast cell degranulation. This can give you a hypersensitized system that keeps you in a negative feedback loop and continues to degrade your health and creates more epigenetic changes and metabolic changes, leading to yet further symptoms of chronic illness, if not eventually breaking your entire immune system, which results in autoimmune diseases, cancers, etc. So, the original cycle remains, but you can see I added in the major histocompatibility 1 and 2. This is an oversimplification to illustrate how this ties together. I mean a seriously oversimplified version. Otherwise, there'd be arrows everywhere and more information than you'd know what to do with. So far, we've had to learn the major histocompatibility complex and the complement system to understand the chronic activation of the immune system. So, mitochondrial damage can trigger the MHC1 complex. Only the MHC3 triggers the complement system, which triggers more mast cells. This, in turn, releases more cytokines. And then the major histocompatibility complex triggers more immune cells. All of these factors, as well as the items in the cycle of illness, are the overall driving factors behind all chronic illnesses. This is why I am constantly preaching about detoxing, fasting, and juicing. We are literally halting all of these vicious cycles taking place in the body and resetting the very biological functions that have been making you sick. We are fixing all of this to the best of our abilities within our genetic code. We have to turn off the vicious cycles taking place, rebuild the microbiome by killing off the bad bacteria, balance the good bacteria, reintroduce the biodiversity of your gut microbiome, which has so many processes linked to it. The microbiome and the gut health are the linchpin that holds the entire cycle of illness together and keeps it moving in a healthy direction, versus an out-of-control, drunken monkey party that's wreaking havoc on your body. We're resetting everything, and if this doesn't help convince you to drop your bad habits and triggers as much as you can, your allergens, change your diet, lifestyle, exercise, lower stress, have a consistent sleeping pattern every single night, this specific cycle will keep going, and you will forever be stuck in a mast cell degranulation or chronic immune response. I don't know how else to illustrate that everything is tied together in the human body, not compartmentalized like modern medicine has been organized. Please keep in mind, this is also an oversimplification of all the processes, but the results are what I have illustrated throughout my videos. The cycle of illness videos, the fasting, the rebuilding through detoxing and juicing, etc. are the way out of chronic cycle of illness. Everything ties to this, and I do mean everything.
Within your body, a unique and elegant story is unfolding. The story begins with the body's natural defenders, specialized cells of the adaptive immune system called T and B cells. In partnership with other immune cells called macrophages, they're constantly scanning for threats, and then jump into action when macrophages present them with a threat. T and B cells have receptors, TCRs and BCRs, which can recognize one or a small number of the millions of antigens to which our bodies are continuously exposed. When a T cell finds its target, it binds and then rapidly multiplies to allow for a powerful attack. It also directs the B cell to produce antibodies. When the battle is over, some of these T cells and B cells stick around, so next time the body sees the same target, they are ready to respond more quickly.
And these are the CD receptors found on B cells and T cells. This is just general information because I know some of you understand this information and some of you actually know which receptors you may be having a problem with. So this can tell you where they're at.
So in today's video, we're going to be talking about B cells and T cells. So they're both white blood cells and they have a role in immune function. So that means that they have a protective role in fighting infections. So there are different types of B cells and T cells, but they all originate from the same cell. And this cell is known as the hematopoietic stem cell, which originates in the bone marrow. So through differentiation processes, this hematopoietic stem cell eventually forms B cells and T cells. Let's talk about B cells first. The main function of B cells is the recognition of antigens. After being made in the bone marrow, they travel via the bloodstream to organs like the spleen, or they're located in lymph nodes, and this is usually where they're concentrated in the body. On the surface of B cells, there are these receptors which are called B cell receptors, and these have a function of binding onto antigens. If you don't know what an antigen is, imagine it's like a small chemical or molecule which is on the surface of harmful bacteria or pathogens, or it doesn't have to be on the surface of a pathogen, but the body recognizes it as a foreign substance. Once an antigen is bound onto the B cell receptors, the antigen is engulfed into the B cell in a process called receptor-mediated endocytosis. The B cell then breaks down the antigen, and the breakdown products are then placed on the surface of the B cell in a special type of molecule called a major histocompatibility complex. And in B cells, it's the second class of major histocompatibility complex which is present. The reason that this occurs is it allows helper T cells, which are also known as CD4 lymphocytes, to recognize and bind onto the major histocompatibility complex class 2. And once it's bound, it releases lymphokines. Lymphokines are important because it causes this B cell with these specific B cell receptors to clone itself via the process of mitosis, so that we have more of these B cells with the same receptors. And it causes the B cells to differentiate into plasma cells. And the main function of plasma cells are to release antibodies. These antibodies that are produced by the plasma cells are specific to the antigen that was initially bound onto the B cell in the first place.
The B cells. B cells protect you against harmful pathogens like viruses, bacteria, and parasites. The two main categories of B cells are plasma B cells and memory B cells.
There are other types of B cells, and they differentiate depending on where they are, whether they're coming out of the bone marrow or the secondary lymphoid organs, like the spleen and the lymphatic system. White blood cells of the immune system are derived and mature in the bone marrow. Once matured, your B cells travel to lymphatic system to include the spleen and lymph nodes. A specific subset of B cells resides within the medulla of the thymus. These cells play a critical role in presenting self-antigens to T cells to help the immune system learn to tolerate the body's own tissues. This process is vital for preventing autoimmune diseases. This is the primary functional relationship between the thymus and B cells to train your T cells. If a T cell strongly reacts to a self-antigen, or basically you, by a thymic B cell, it is signaled to undergo apoptosis, or cell death, preventing it from attacking the body's own tissue later on. So the thymus is the schoolhouse for the T cells.
and the B cells help train them. B cells consist of pre-B cells. These are called progenitor B cells or B cell precursors. These are earlier precursors to the naive B cells. These develop in the bone marrow and express a surrogate B cell receptor. Naive B cells, aka the pro-B cell or progenitor B cell, these are immature B cells that have not yet encountered an antigen, and they are found in the bone marrow. B1 cells, these are a subset of B cells that are found in the peritoneal cavity and mucous membranes. They produce antibodies against polysaccharides and lipids. Marginal zone B cells. These are B cells that are located in the marginal zone of the spleen. They are specialized in responding to blood-borne pathogens. Follicular B cells. These are the most common type of B cell. They develop in the spleen, and they are found in the follicles of the lymph nodes and produce antibodies against the proteins. Memory B cells. These are B cells that have previously encountered an antigen and are able to respond more quickly to future infections. Plasma cells, these are mature B cells that produce large amounts of antibodies. They are formed with naive B cells and activated by an antigen. Regulatory B cells, these are a subset of B cells that suppress the immune response. They play a crucial role in helping T cells self-correct by acting as an antigen-presenting cell to basically teach T cells that they are maturing in the thymus. If the T cells react or overreact to self, meaning they can't tell the difference between what's you and what's an actual foreign invader, then they are eliminated. This is the process that takes place that helps maintain immune tolerance. If this process doesn't work, it can seem like your immune system is completely freelancing. And they also help to prevent autoimmune diseases. And now remember, the major histocompatibility complex and the complement system all plays into this. Okay, so pre-B cells, aka progenitor B cell or B cell precursor. These are early precursors to the naive B cell. Immature B cells that are still developing before they become fully functional immune defenders. These develop in the bone marrow and express a surrogate B cell receptor. The bone marrow is the training ground for immune cells. They are a crucial checkpoint in B cell development, making sure that they can successfully produce antibodies before they mature. They make heavy chain of antibodies. Sorry, I'm getting tongue-tied over here. Which are part of the defense weapon. Once they make it past this stage, they move forward to complete the light chain and become fully developed B cells. If pre-B cells don't develop correctly, they can't produce proper antibodies, which weakens your immune system. They help ensure that only properly functioning B cells make it into the bloodstream. Naive B cells, otherwise called as pro-B cell or progenitor B cells. These are immature B cells that have not yet encountered an antigen, but they are already ready to fight. They are created in the bone marrow and then move to circulate in the blood and lymphatic system. They wait for their first mission, encountering a pathogen. They carry unique antibody receptors on their surface, allowing them to recognize specific patterns. They haven't been activated yet, meaning they haven't encountered the enemy or the antigen that they were designed to fight. Once they meet their magic antigen, they become activated B cells, transforming into plasma cells or memory B cells. And the plasma cells are antibody factories, and the memory B cells are long-term immunity. They ensure the immune system is prepared to recognize and attack new threats. They provide the first step in learning immunity, setting the stage for stronger, faster responses in the future. B1 cells. These are the CD5+ B cells, also referred to as innate-like B cells, but different from marginal zone B cells. And remember, marginal zone B cells are in the spleen. They are a special type of B cell that provides quick, natural immune responses, especially against the common bacterial infections. They are a subset of B cells that are found in the peritoneal cavity, which is around the abdominal organs, the pleural cavity, which is around the lungs and the mucous membranes, and in circulation. They produce natural antibodies without needing prior exposure to an invader.
sometimes called innate like B cells because of their ability to produce natural antibodies without their prior activation, bridging the gap between innate and adaptive immunity. They respond quickly to threats, unlike regular B cells that need activation. They are T cell independent, meaning they don't need help from T cells to get activated. They produce antibodies against carbohydrates on bacteria, as well as polysaccharides and lipids to a point. It's more of an indirect relationship, which makes them key defenders against certain infections like strep, the flu, and meningitis. They provide early defense, especially against bacterial infections. They bridge the gap between the innate and adaptive immunity, offering quick but less specific protection. They are self-renewing, meaning they can maintain their population without constant replenishment from the bone marrow. So, B1 cells play a crucial role in the immune response against polysaccharides, but have a limited direct role against lipids. B1 cells and polysaccharides. B1 cells are especially important for recognizing bacterial polysaccharides, which are commonly found in bacteria capsules. It is the protective outer layer of bacteria. They produce natural IgM antibodies that target these polysaccharides, providing early, rapid defense against bacterial infections. Examples of bacteria with polysaccharide-rich capsules that B1 cells help fight include strep, the flu, and meningitis. B1 cells and lipids. B1 cells do not primarily target lipids, but they do contribute indirectly to immune responses involving glycolipids, molecules with both carbohydrate and lipid compounds. Some glycolipid antigens, such as those found in certain bacteria, like mycobacteria, can trigger B1 cell responses. However, lipid-specific immune responses are mostly handled by other immune cells, like invariant natural killer T cells, which specialize in recognizing lipid antigens and presenting it to the CD1 molecules. B1 cells are highly effective against bacteria polysaccharides and produce antibodies that neutralize bacteria with polysaccharide-rich capsules. Their role in lipid immunity is limited, but they may still participate in responses to glycolipid antigens. Other immune cells, like the invariant natural killer T cell, specializes in lipid recognition. Okay, so all I really want you to remember in this one, besides the rest of this information. So if you have leaky gut, for example, and you're suffering with a lot of abdominal pain, a lot of abdominal burning, and it doesn't feel like it's in your gut, it's outside of your intestinal tract, this is what's getting activated. If you're having spleen pain, your B cells are activated. If you're having lymph node pain, your B and T cells are activated. So you want to take this seriously because it has long-term consequences. And speaking from experience, you get these cells out of control, oh man, you're going to be miserable. Marginal zone B cells, also known as innate like B cells, because they act as a bridge between the innate, fast and general, and adaptive, specific long-term, immunity. They are also known as splenic marginal zone B cells. These are the B cells that are located in the marginal zone of the spleen, a key area where blood is filtered and pathogens can be detected. They are specialized immune cells that are the first responders against bacteria and the viruses in the blood. They quickly recognize and respond to harmful invaders, especially bacteria with polysaccharide or sugar-based coating. They provide quick protection against bloodstream infections, especially from the bacteria that avoid detection by other immune cells. This can activate rapidly without needing T cell help, making them part of the fast-acting immune defense. They produce IgM antibodies, which help neutralize threats before they spread. They also act as messengers presenting antigens, or foreign molecules, to the other immune cells to kickstart a stronger immune response. They help bridge innate and adaptive immunity, ensuring a fast response and allowing a more targeted immune reaction to develop. Dysfunctional marginal zone B cell response can contribute to immune deficiencies or autoimmune diseases. Follicular B cells, or B2 cells. These are the most common types of B cell.
They develop in the spleen, and they are found in the follicles of the lymph nodes and produce antibodies against proteins. When not circulating through the blood, it is in the lymphoid follicles of secondary lymphoid organs, which is the spleen. They live in lymph nodes, spleen, and other lymphoid tissues inside specialized areas called follicles. They are responsible for making highly specific antibodies and providing long-term immunity. They are responsible for generating the majority of high-affinity antibodies during the infection. They detect and respond to harmful invaders like bacteria and viruses. They actually need T cells to activate them to become fully activated. And once they are activated, they transform into either plasma cells, which produce large amounts of antibodies to help fight the pathogens, or memory cells, which provide long-term protection against the reinfection. Memory B cells. These are B cells that have previously encountered an antigen and are able to respond more quickly to future infections. Now, mature B cells in general, including those in the peripheral blood, typically have a half-life of 5 to 6 weeks, with a small percentage being short-lived and lasting only a few days. Memory B cells, however, which are crucial for long-term immunity, can live for years, potentially even decades, in your body and in your bone marrow. Vaccines actually help exploit this process. Dormant B cells arise from B cell differentiation. These cells travel through the body and trigger a faster, stronger antibody response when needed. The amnesic secondary antibody response, which helps the immune system remember past threats. How it works. If they come across the same antigen, say like a harmful substance, that previously activated their parent B cell, they quickly respond. Their parent B cells recognize the same antigen, so they know exactly what to attack. Okay, so how are they created? They can be activated with the help of T cells through two processes: the extrafollicular response, which is a rapid, early response, or the germinal center reaction, a more refined, stronger immune response. They can also be activated without T cells through a separate process involving B1 cells. Plasma cells. These are mature B cells that produce large amounts of antibodies. They are essential for humoral response, and remember that kills the pathogens, not the cell. They are formed with naive B cells and are activated by antigens. They include plasmablasts, and these are acting as a precursor cell that eventually differentiates into fully mature cells or fully mature plasma cells. They rapidly produce antibodies during and early immune response. Plasma cells are the more mature, long-lived antibody-secreting cells and from plasmablasts. Plasma cells are responsible for sustained antibody production over a longer time. So they can be activated by T cells, and those particular plasma cells are needing the T cells to activate them, and then there are other plasma cells where they can activate themselves. Lymphoplasmacytoid cells. A cell with a mixture of B lymphocytes and plasma cells. Morphological features that is thought to be closely related to a subtype of plasma cells. In the diagram here, you can see how the pre-B cells are coming out of the bone marrow, and then it turns into the B cell, and then it gets to the lymph nodes where it becomes a germinal center. And through this next process, what can wind up happening is there's a genetic mutation, and then it changes the cells. You can see that they were blue at first, and the genetic changes were yellow. And then what they do is they combine together, and that's how you get lymphoplasmacytic cells. The yellow and blue are now part of the same cell. You have the yellow and the blue in the same cell. So that's essentially what this is saying, and this can wind up being a slow-burning cancer. This cell type is found in pre-malignant and malignant plasma cell dyscrasia. Dyscrasia is a non-specific term that refers to a disease or disorder or imbalance of the blood. Before modern medicine, it meant an imbalance of the four body fluids: blood, bile, lymph, and phlegm. The lymphoplasmacytoid cells are associated with the secretion of IgM monoclonal proteins. Waldenstrom's macroglobulinemia is one of those, Waldenstrom's is one of those cases where your body's making lymphoplasmacytoid cells, and it's kind of like a very slow burning cancer. The presence of lymphoplasmacytoid cells is also a warning sign, or a warning sign for a type of lymphoma, and Waldenstrom's is a type of lymphoma. Okay, regulatory B cells. These are a subset of B cells that suppress the immune response, so it keeps your immune system from overreacting and regulates that. They play a crucial role in helping T cells self-correct by acting as antigen presentation cells to basically teach the T cells that are maturing in the thymus. So the B cells go to the thymus, they hand antigens and test basically the T cells to know the difference between what is you and what is a foreign invader. So the thymus is kind of like a schoolhouse for T cells, and these regulatory B cells are going to the thymus, kind of acting like teachers to the T cells. If the T cells react or overreact to self, then they are eliminated. This is the process that takes place that helps maintain immune tolerance. This is what keeps your immune system from reacting to everything. If this process doesn't work, it can seem like your immune system is completely freelancing through your body. Regulatory B cells also help to prevent autoimmune diseases because, like I said, regulatory B cells suppress an overreaction of the immune system. They stop the expansion of pathologic pro-inflammatory lymphocytes through the secretion of interleukin 10. Interleukin 10 is known as the human cytokine synthesis inhibitory factor. So basically, the interleukin 10 is the chemical or the cytokine that gets released to help prevent your immune system from going haywire and overreacting. This helps prevent damage to you and maintains normal tissue homeostasis. They are produced by CD4, those are your helper T cells, and CD8 T cells, those are your killer or also known as cytotoxic T cells. And active B lymphocytes, monocytes, macrophages, and keratinocytes. As an anti-inflammatory cytokine, interleukin 10 down-regulates the expression of T helper cell cytokines, the major histocompatibility class 2, and co-stimulatory molecules on the macrophages. So the interleukin is what's helping to keep your T cells, your major histocompatibility complex, and the way it responds by regulating it so that it doesn't cause you harm. Regulatory B cells also stimulate the Fc gamma receptor expression. This is a protein that helps the immune system recognize and respond to pathogens. It is a receptor for the IgG, which is a protein that helps immune system recognize and respond to pathogens. IgG can also cause autoimmune conditions, be behind it, as well as allergic reactions. They are found on macrophages, neutrophils, and B lymphocytes. They're also part of phagocytosis. And remember, this helps the body destroy microbes and infected cells. Now, polymorphisms in the Fc gamma receptor can affect the ability of the body to bind the IgG and phagocytose, which means it makes it hard for the immune system to detect cells that are infected without this receptor working properly, because remember, the receptors like hold out a flag that says, hey, immune cells, I'm right here. And if they're not doing that, then that means that your immune system might not respond to the pathogens the way it's supposed to. So the B cells help keep the immune system balanced. And one of the ways they do this is by increasing something called the Fc gamma responders. And the Fc gamma responders is like a special sensor that helps immune cells recognize and respond to threats, especially when antibodies like IgG have tagged a problem. They are found on macrophages, neutrophils, and B cells, which are the frontline fighters of your immune system. Okay, let's take a closer look at this. I'm sure this is all very confusing right now, but I'm about to clear it up for you. I hope. Okay, so we're talking about B regulatory cells and the Fc gamma receptors, okay? The B regulatory cells regulate these receptors, and these receptors help the immune system recognize and bind to pathogens. To do this, they have to recognize antibodies. Okay, so then there's IgG,
IgA and IgE antibodies that form different roles in the immune system. And those of you who have a lot of health issues are probably very aware of IgG, IgE, or IgA. And these bind to very specific Fc receptors. So there's all different kinds of Fc receptors. So let's look at the IgG, all right? The antibody IgG is the most abundant antibody in the bloodstream and other body fluids. It provides long-term protection against infections, so therefore they have memory. It remembers past infections. They neutralize toxins and they activate the complement system, which helps destroy the pathogens. And remember, we were talking about the complement system and the major histocompatibility complex. This fits into all these receptors. That's what creates the chain that links everything together. So the IgA antibody is found in mucosal secretions like saliva, tears, respiratory, and intestinal fluids. It guards the body's entry points for pathogens or germs, etc. It acts like a barrier, preventing pathogens from attaching to and invading mucosal surfaces. Now, the antibody IgE is primarily involved in allergic reactions and defense against parasites. They usually present low concentrations in the bloodstream. These also bind to mast cells and basophils. This releases histamine and other chemicals that cause allergy symptoms. So the easiest way to remember this is IgG is pathogens, IgE is parasites and allergic reactions. Now, when it comes to B regulatory cells, they work very closely with IgG antibodies, and they can combat infection in a couple of ways. The first way that an IgG can work is by binding to the pathogen directly. They use the C1 from the complement system to do this. This will trigger the complement classical pathway. You can see my little drawing of the bacteria or pathogen with my little receptors, and then next to it there's like a block that says C1. That's part of your complement system, and your complement system is what is reacting to the bacteria or pathogen, okay? The other way that IgG can combat infection is by the immune cell attaching to the pathogen, and it calls more phagocytes, which are your macrophages, your dendritic cells, and your neutrophils. The immune cell then binds to the pathogen using the Fc gamma receptor on the bacteria. So either your complement system is attaching to the bacteria or pathogen, which, remember, your complement system is like your first responders. And then the other way is when your immune cell directly attaches to the bacteria or pathogen. One way to kind of look at it as the complement system that attaches to the bacteria or pathogen is kind of like the roundabout way to get your immune cells involved. Whereas the number two, your immune cell is directly involved and goes right after the bacteria or pathogen. So they're using the Fc receptors to either connect the complement system to the bacteria or pathogen, or the immune cells to the bacteria or pathogen. When the immune cell attaches to the pathogen, it winds up covering the pathogen with the IgG antibodies, and this is what makes phagocytosis work more efficiently. And remember, phagocytosis is when the immune cells engulf the bacteria and eat it, and then spits out tiny bits and pieces, or I like to call the crumbs, to the end of those receptors to alert other immune cells to kind of be on the lookout because there's a pathogen in the body. Are you following so far? I hope so, because I don't know a better way to explain this. Okay, so this picture is not as good, but bear with me. Immune cells cannot go into the cells to fight pathogens, okay? So that's when the humoral response kicks in. And remember, the B cells do this. The pathogens get broken up via phagocytosis, and the pieces and parts of it are displayed on the outside of the cell to alert the immune system. And proteins of pathogens may also have left signs of itself on a cell surface when it got into it and infected the cell. So if you think of a pathogen trying to get into one of your cells, drilling a hole or eating through and getting into the cell to infect it, it could have left pieces or remnants of itself on the outside of the cell. The body will then make IgG against those pathogen proteins to help the body identify that the cell is infected. Now, IgG is primarily produced by the plasma B cells. When a B cell encounters an antigen, like say IgG,
That's a substance that triggers an immune response. It can become activated. Activated B cells differentiate into plasma cells, which are specialized for antibody production. So plasma cells are what's making all of these IgG. And these plasma cells then begin to secrete IgG antibodies, which are specific to the encountered antigen. So they're very specific. So in our little example of the little green goblin pathogen here, our body secretes the IgG through the plasma cells to attack specifically the green goblin. Nothing else. It makes antibodies to attack the green goblin pathogen. So when the body makes IgG against those pathogens, they help the body to identify that the cell is infected. So let's say a pathogen has made it into the cell. Here are some body cells, and we make antigens to fight a virus. And the virus or pathogen is hiding inside the cells. So the body makes antibodies to combat the pathogen or virus, which is also called antigens. But antibodies don't go hunting for pathogens or viruses or antigens inside the cells. Antibodies exist in your humerus or humoral immune response. They are in the fluid inside the cells. So that body cell that is infected by pathogens or viruses, it may display some of those pieces on the infected cell. Viruses also like to replicate, so it may start leaving pieces on the outside of the cell or trying to escape the cell to find other cells to infect. If we make IgG against those proteins, or in our example, the green goblin pathogen, the immune system can identify which cells are infected. And in this example, the Fc receptors are what are used to bind to the IgG for the natural killer cells to go through and kill off the cell and kill the infection. So when it comes to mast cells, you are dealing with IgE versus IgG. The IgE binds again to those Fc receptors. And remember, like I said earlier, there's all kinds of different Fc receptors. As of right now, we're just calling them Fc receptors because I don't want to confuse you with all these different names for the different types of Fc receptors and what cells they live on. So for now, just think Fc receptors to make it easier to understand. So we've already said that pathogens stimulate IgG. Well, they also stimulate IgE, and IgE is the stuff allergic reactions are made of. Pathogens stimulate IgE, parasites stimulate IgE, and IgE will bind to the surface of mast cells. The Fc receptors are high affinity, and that's an antibody that binds to the cell first. It's kind of like priming the cell. If a pathogen comes near the mast cell uses the IgE and cross-links to the pathogen. So in our little picture here, the sideways worm-looking thing is our pathogen, and the mast cell is basically using the IgG, those are those green lines, and basically kind of reaching out to the pathogen, and that is called IgE cross-linking. So the IgE is already on the mast cells, they're already primed, so when a pathogen kind of comes into its territory, the mast cells automatically bind to it using the IgE cross-linking. And because of this, then the mast cells degranulate, and this can cause allergic and immune responses. And as we've previously learned, mast cells release a host of toxic chemicals that include histamine, cytokines, growth factors, interleukins, prostaglandins, etc., etc. Basophils and eosinophils can also carry the IgE and cross-link to foreign invaders, causing degranulation. And in order to do this, they're all using Fc receptors to bind. Alright, why did I teach you all this? The Fc receptors are vital to the immune system working properly. Whether it be IgG, IgA, IgE, the immune system needs the Fc receptors for binding of immune complexes to kill diseased or infected cells, damaged cells, etc. If there is a mutation in the Fc receptor, then the immune system can't do its job efficiently. It's kind of like showing up to a fight with both of your hands tied behind your back. So this can have major repercussions for the immune system and your health. And B cells help regulate these Fc receptors. So this process with the B regulatory cells and the Fc gamma receptor, it helps kill infections and it helps the body to destroy tumor cells. It presents antigens to T cells, like showing the enemy's photo to the army. It regulates the immune system so it doesn't overreact. And it helps clear out leftover immune debris, your immune complexes.
So mutated Fc receptors are going to have a big impact on how your immune system works. This can cause your immune system to seem like it's going feral and cause autoimmune diseases such as rheumatoid arthritis, amongst many others. Now, this can affect your mitochondria in a very significant way, and not in a good way, because of the secretion of reactive oxygen intermediates, which, remember, reactive oxygen species is kind of like a rusting in your cells. High or dysregulated interleukin 10 production is associated with autoimmunity and can represent for disease susceptibility and or severity. So if you have problems with your B cells, you have mutations with the Fc receptors, which then makes your immune system kind of freelance all over the place, and then because of the degranulating cells and the immune response, is going to lead to autoimmune disorders, and it's going to affect the severity of how bad you get them. Sjogren's syndrome, Graves' disease, autoimmune lymphoproliferative syndrome, lupus, cancers, and cardiovascular diseases like heart failure fall under this category. Regulatory B cells also regulate the interleukin 35, and that's responsible for response in reducing inflammation. It is part of the interleukin 12 family of interleukins, and it is secreted by regulatory B cells and the T cells. It's also secreted by regulatory T cells. It suppresses the proliferation of T helper cells, Th1 and Th17, and we'll get into the T cells in a little bit and you'll understand what that means. It blocks inflammatory signaling pathways, and it regulates balance of the T cells and T regulatory cells. This can induce regulatory B cells that suppresses autoimmune diseases. It's also involved in development of inflammatory diseases like rheumatoid arthritis, and it can help control immune-related disorders like autoimmune diseases, infectious diseases, and cancer. So it's really important that your regulatory B cells and T cells are letting out the correct amount of interleukin. Too much or not enough is very harmful to your body. It also may help alleviate neuropathic pain to a point if you get these under control. It'll also release transforming growth factors, TGFB, and it's a multifunctional cytokine that belongs to the transforming growth factor superfamily. All white blood cell lineages produce tumor growth factor Bravo. It's a protein that regulates cell growth, tissue repair, and the immune system. It is produced by almost every tissue and cell type. It plays in many processes, including embryonic development, wound healing, and cancer. It maintains tissue homeostasis. It inhibits expansion of some immune system components, so it kind of helps the immune system from getting too far out of whack, but it can only do it to a certain degree. It promotes immune tolerance, and that is where it helps regulate your immune system so your immune system doesn't go crazy and start attacking everything. It helps repair injuries, controls cell proliferation, which means when your B cells come into contact with a pathogen, they start proliferating, or I suppose they basically start cloning themselves and start making many copies of themselves. And this helps control that so it doesn't get out of hand. The acidic body can make these processes worse. So it's very important to keep your body as alkaline as possible. In disease, transforming growth factor Bravo promotes inflammation, contributes to fibrosis and cancer, contributes to resistance to cancer treatments, contributes to autoimmune diseases, and contributes to cardiovascular diseases like heart failure and cataracts. Transforming growth factor Bravo in cancer inhibits cellular transformation and prevents cancer progression in early stages. So if you're not doing what you can to get healthy and these processes keep running amok because you're constantly giving it triggers, infections, or whatever the case may be, it later becomes a tumor promoter, facilitating cancer cell growth and metastasis. So again, it's super important that you keep your immune system and your B regulatory cells under control.
Remove your triggers. Get your stress under control. Exercise and get good night's sleep and a good diet. So you need to make sure you're removing your triggers, getting your stress under control, exercising, sleeping a consistent amount of sleep every night, because again, that helps your methylation. And then on top of that, you want to make sure you're getting a good diet and exercise, because you have to take care of your immune system, and those are how you do it. Because if you're not changing your habits that are making you sick, you run into all these additional problems. So regulatory B cells have a big job to do, and it's important to keep your immune system in balance as much as you can. One type of B cells can alter how the other B cells also work, and it also alters how your T cell works. So if you are noticing that you have a lot of lymph node pain, please, please, please take this seriously and start eating a balanced diet or fasting if it gets bad. Managing your stress levels. Avoid alcohol or no smoking or vaping. Quit if you do. You need 7 to 8 hours of sleep per night. Moderate exercise, approximately 150 minutes per week. And keep good hygiene. This actually affects your B cells. Testing can tell you what your B cells are doing. 1,000 to 4,800 lymphocytes per microliter of blood is normal. 10 to 20% of your lymphocytes are B cells, and having chronic high or low B cell may mean you have a condition or disease. And a standard CBC test can measure this. Problems with your B cells, the autoimmune diseases tend to be lupus, MS, rheumatoid arthritis, and type 1 diabetes. If your B cells are out of control, it can cause a lot of blood cancers, such as acute and chronic lymphocytic leukemia, non and regular Hodgkin's lymphoma, B cell lymphoma, multiple myeloma, and Waldenstrom's macroglobulinemia. And remember, that last one is a very slow growing cancer. If you have spleen pain, pancreas pain, and lymph node pain, please take this seriously. If you are experiencing these painful swollen lymph nodes, it's likely that your B cells are reacting within the lymph nodes, producing autoantibodies against your body's own tissues, which can lead to inflammation and pain. If B cells are chronically active, it can lead to the development of autoimmune diseases where the immune system mistakenly attacks the body's own tissues due to the production of autoantibodies against yourself, potentially causing inflammation and damage in various organs depending on the target antigen. Conditions like systemic lupus, rheumatoid arthritis, and MS are examples of this phenomenon. If you have an overactive immune system, your body may react to additional protein added to it, potentially triggering an inflammatory response because your immune system might misinterpret the new protein as a foreign invader, leading to symptoms like inflammation, pain, and great fatigue, depending on the specific autoimmune condition you have. It's important to consult your doctor before making significant dietary changes, especially if you have an autoimmune disorder. B cells play a role in helping T cells self-correct by acting as antigen-presenting cells, presenting self cells to the T cells during their development or training in the thymus. This allows the immune system to eliminate any T cells that strongly react to cells that are part of you. This process is the key to help maintaining immune tolerance. If both your B and T cells are not performing properly, adding more protein can make your symptoms worse because it's proteins they attack, not knowing which is you and which is the foreign invaders they're supposed to be attacking. So if your lymph nodes are hurting or eating protein makes you feel worse, especially if you have spleen pain, pancreas pain, pain in the right side as well, you want to take this seriously. This is a huge red flag that you need to address right away before developing autoimmune disorders, cancer cells begin to form, and possibly invade the immune system from both aberrant processes and the inability to properly determine what is friend or foreign. So this diagram shows how the main types of regulatory B cells interact with your other immune cells and what they do. I am not going to go in depth into this today because I do not want to probably confuse you any more than you probably already are. So please pause the video to look this over, or you can find this in the cycle of illness immune system slides.
It's that time again for How Is It All Connected? Okay, the part you've probably been waiting for the most, that makes the most sense. So, the B cells and the BH4 cycle. BH4 is needed for both B cells and T cells to work properly. So if you have the MTHFR 1298, you might want to take this a little more seriously because you're going to have a few more problems in this department if you don't take care of your immune system and cut out your triggers. Without enough BH4, B cells may not mature correctly, meaning fewer antibodies are produced, which means extra toxins, viruses, infections, etc. go up. Low BH4 levels can lead to immune fatigue, making it harder for B cells to fight infections. B cells and NOS, the nitric oxide system, that is part of the urea cycle. B cells rely on nitric oxide to communicate and function efficiently. So if you have NOS, you might be hampered in this department. So you're also going to have to take extra care to keep your immune system healthy. NOS, the enzyme that makes nitric oxide, needs BH4 to work correctly, and we went over this in great detail in the methylation video. If NOS is not functioning properly, B cells become inflamed instead of fighting off infections properly. Too much nitric oxide can make B cells overreact, leading to autoimmune issues where the immune system attacks healthy cells. B cells and lymph nodes. Lymph nodes are the meeting place where B cells get activated. When an infection happens, B cells inside the lymph nodes multiply and start producing antibodies. If the immune system is overactive due to too much nitric oxide or oxidative stress, so if you're drinking sodas, smoking, doing drugs, alcohol, or severe chronic stress, etc., this is going to have a huge impact on that. B cells may start mistakenly attacking the body's own tissue, leading to autoimmune diseases. B cells and the spleen. The spleen is an important training ground for B cells. Marginal zone B cells live in the spleen and act as first responders to blood-borne infections. And that's why I'm saying if you have spleen pain on top of lymph node pain, you need to take this seriously because you have something going on inside of you. If this is you, then fasting can help shut down these aberrant processes and help the body catch up and heal the infection or whatever it is you have going on. If you do have this happening, please see your doctor. If the spleen isn't functioning well, B cells might miss certain infections, leaving the body more vulnerable. B cells and oxidative stress in mitochondria. B cells need mitochondria to produce energy so that they can multiply and produce antibodies. When oxidative stress is too high, again, sodas, smoking, drinking, not sleeping, and way too much stress also raises oxidative stress. The mitochondria don't work efficiently and B cells struggle to function. Too much oxidative stress can lead to B cell dysfunction, meaning fewer antibodies and a weaker immune response. In autoimmune diseases, oxidative stress can cause B cells to go rogue, attacking healthy tissues instead of the infections. So B cells activate T cells, and in turn, the T cells activate the B cells so that they can produce antibodies. When a B cell recognizes a threat, like a bacteria or virus, they need permission from helper T cells, those are your CD4+, to go into full attack mode. Once activated, B cells become plasma cells, which are antibody-producing factories, or memory B cells, which remember the threat for faster response next time. A balanced BH4 and NOS equals healthy T and B cells and strong immunity. Too much oxidative stress equals weakened or overactive B cells, leading to immune dysfunction. And dysfunctional lymph nodes and spleen equals poor B cell activation, leading to chronic infections or autoimmune disease. BH4 and NOS regulate T cells and B cells, ensuring they work properly and don't overreact. T cells activate B cells in the lymph nodes and spleen, helping them create antibodies. If oxidative stress is too high, mitochondria can't produce enough energy, and B cells can't fight infections properly. If NOS isn't balanced, B cells might get confused and start attacking healthy cells, and that's an autoimmune reaction. Without enough BH4,
both B and T cells can become sluggish, leading to weaker immunity and chronic illness. And remember, if you're too acidic, this is greatly going to affect how these cells work. So this delicate balance is what keeps your immune system strong but not overreactive. When part of the cycle is off, say your mitochondria, your methylation, your gut microbiome, B cells, T cells, and overall immunity can definitely suffer.
So in today's video, we're going to be talking about B cells and T cells. So they're both white blood cells and they have a role in immune function. So that means that they have a protective role in fighting infections. So there are different types of B cells and T cells, but they all originate from the same cell. And this cell is known as the hematopoietic stem cell, which originates in the bone marrow. So through differentiation processes, this hematopoietic stem cell eventually forms.
Remember, B cells show antigens and present them to the helper T cells, which activates them. And then in turn, the helper T cells activate the B cells. And our helper T cells are the CD4.
Now let's talk about T cells. We just mentioned one type of T cell called the helper T cell, also known as CD4 T cells. And just to recap, it binds onto the second class of major histocompatibility complex, which is a molecule located on the surface of B cells, and this binding causes the release of lymphokines. Other types of T cells include the cytotoxic T cell, also known as CD8 T cells. In the case of cytotoxic T cells, or CD8, imagine you have a cell infected by a virus. The cell is now infected and it contains viral DNA and viral mRNA. Because of this, the virus is now making viral proteins and peptides. Since all cells with a nucleus have major histocompatibility complex class 1 on their membrane, these viral proteins that the infected cell is making is presented by the class 1 major histocompatibility complex molecule on the surface of the infected cell. So even though that sounds very confusing, imagine the infected cell is kind of giving a warning signal by showing the infected proteins on its membrane. The cytotoxic CD8 cells have T cell receptors on their surface that recognize the peptide on the surface of the infected cell is not normal, so it binds onto it, and when it binds, the T cell either secretes cytotoxins like perforin or granulysin, which initiates programmed cell death in a process called apoptosis, or once the binding occurs, the T cell expresses a protein called Fas ligand or FAS ligand, which binds onto the FAS molecule present on the infected cell, and this forms the death-inducing signaling complex, and eventually we have apoptosis of the cell. We also have memory T cells, and there are different types, but the main thing that you need to know is that they are able to replicate very fast and initiate their response very rapidly if they've encountered the antigen before. The final point that we're going to make is that both B and T cells are part of the body's natural adaptive immunity, which means they're a part of a specialized network of cells that specifically respond to pathogens and specifically fight off infections.
Okay, that's a great entry for T cells. So let's take a slightly closer look at it. T cells, or T lymphocytes, are a type of white blood cell that plays a crucial role in the adaptive immune system. It is also the main component of the adaptive immune system. Think of it as like a specialized smart system that's constantly monitoring for threats. Once it detects an intruder, your adaptive immune system builds a customized defense to fight it. Each T cell is unique in that it's designed to fight only one type of intruder. Once the threat has been identified, it locates the specific T cell designed to defeat it and recruits that specific T cell for battle. Now, we talked briefly about the different kinds of T cells. There is the CD4 helper T cells, These cells coordinate the immune response by activating other immune cells. They act as commanders, directing other immune cells on how to respond. They release signals, or cytokines, that boost or regulate the immune response. They have the CD4 receptor, that's why they're CD4 plus T cells, and they do not kill cells directly. Instead, they activate other cells in your immune system how to coordinate an attack against the invaders. It also signals cytotoxic T cells, those are your killer T cells, B cells, and macrophages. The other type of T cell is the CD8+, or the cytotoxic or killer T cells. These cells directly kill infected and cancerous cells. They act as killers, targeting and destroying infected or cancerous cells. They use toxin proteins to break down harmful cells and stop the spread of infection. These proteins are called perforin and granzymes. Then there's the regulatory suppressor T cells called TREGs. These cells suppress the immune response to prevent an overreaction. They kind of act like a peacekeeper, preventing the immune system from attacking healthy tissues. They also help prevent autoimmune diseases by calming overactive immune responses. So this also helps with your immune tolerance. Memory T cells is another type of T cell, and they are long-lasting immune cells that protect against pathogens like virus and bacteria. And we're talking years, possibly decades. Okay, there's 3 types of memory cells, and this is all we're going to discuss about them, but there's the central memory cells, the effector memory cells, and the resident memory cells. The central memory cells stay in the lymphoid tissue and recognize antigens. The effector memory cells have slightly higher functionality and travel through the body. When a naive T cell encounters a pathogen, it proliferates and differentiates into an effector T cells. The effector T cell migrates to the inflamed tissue and kills infected cells. After the pathogen is eliminated, most effector cells die, but a small number of memory cells remain. And resident memory T cells persist in epithelial barrier tissues like the skin, lungs, and gastrointestinal tract. Memory T cells can quickly respond to a pathogen they've encountered before. Memory T cells are also critical for effective immune responses to vaccines and especially important during immunosuppression. Interleukin 15 is the cytokine most critical for CD8 memory T cell homeostasis and for expansion following an antigen activation. Interleukin 7 and interleukin 15 can rescue memory CD8+ killer T cell homeostasis. Don't worry, I have charts for the interleukins and for the CDs coming up. T cells help protect the body from infections, tumors, and foreign invaders. T cells are produced in the bone marrow and mature in the thymus gland. And remember, your B cells come to present self-antigens so that it trains the T cells the difference between you and the foreign invaders. And if the T cell overreacts as it's training it in the thymus schoolhouse, then it's going to wind up self-destructing because the body doesn't want a kamikaze T cell. They develop in the thymus, hence the T in T cells, and then circulate in the blood, lymph nodes, and tissues ready to respond. They can trigger B cells. Mature B cells help train the T cells when it comes identifying self-virus versus non-self. If T cells overreact to self, then they are killed off to help maintain T cell homeostasis and prevent autoimmunity. When T cells encounters an antigen, it becomes activated and releases cytokines. And remember, cytokines are those signaling molecules that stimulate other immune cells such as B cells and cytotoxic, aka killer T cells. Let's take a look at a short video to help you put the picture together.
There are 2 major classes of lymphocytes involved with specific defenses: B cells and T cells. Immature T cells are produced in the bone marrow, but they subsequently migrate to the thymus, where they mature and develop the ability to recognize specific antigens. T cells are responsible for cell-mediated immunity. B cells, which mature in the bone marrow, are responsible for antibody-mediated immunity. The cell-mediated response begins when a pathogen is engulfed by an antigen-presenting cell, in this case, a macrophage. After the microbe is broken down by lysosomal enzymes, antigenic fragments are displayed
with MHC molecules on the surface of the macrophage. T cells recognize the combination of the MHC molecule and an antigenic fragment, and are activated to multiply rapidly into an army of specialized T cells. One member of this army is the cytotoxic T cell. Cytotoxic T cells recognize and destroy foreign cells in tissues or virus-infected cells. Another T cell is the memory cytotoxic T lymphocyte, which remains in reserve in the body. If sometime in the future, these T cells re-encounter this specific antigen, they will rapidly differentiate into cytotoxic T cells, providing a speedy and effective defense. Helper T cells coordinate specific and non-specific defenses, in large part by releasing chemicals that stimulate T cell and B cell growth and differentiation. Suppressor T cells inhibit the immune response so that it ends when the infection has been controlled, whereas the number of helper T cells increases almost at once. The number of suppressor T cells increases slowly, allowing time for an effective first response.
Okay, like we just watched, an antigen-presenting cell, or APC, like a macrophage, locates evidence of an intruder and attaches it to a structure called the major histocompatibility complex. T cells can't recognize evidence of an intruder unless it's attached to a major histocompatibility complex. CD8 cytotoxic killer T cells can only bind to MHC1, and CD4 helper T cells can only bind to MHC class II. There are 25 million to a billion different T cells in your body, and each one has a unique T cell receptor that can fit with only one kind of antigen. So T cells have receptors called TCRs, or a T cell receptor, that can bind to specific antigens or foreign molecules. When a T cell encounters an antigen, it becomes activated and releases cytokines, which is a signaling molecule which stimulates the immune cells, such as B cells, cytotoxic or killer T cells, and macrophages. Let's take a closer look with this video.
At the interface between a dendritic cell and a T cell, two types of signals are transmitted that lead to a T cell activation. The primary signal hinges on the T cell receptor's interactions with the antigen MHC complex on the dendritic cell. T cells carrying the CD4 coreceptor bind to MHC2 on dendritic cells. In contrast, T cells carrying CD8 bind to MHC1 on dendritic cells.
However, there is no immune response without the secondary signal to activate the T cells. So when a T cell encounters an antigen, it becomes activated and releases cytokines, or signaling molecules that are pro-inflammatory, that stimulate immune cells such as B cells, cytotoxic T cells, and macrophages. Cytotoxic T cells directly kill infected cells by releasing proteins called perforin and granzymes. Now, T cell hypersensitivity is usually delayed, but due to lower BH4 or too much oxidative stress or mitochondria damage can make your T cells very hyperactive. So, if you have the MTHFR 1298, which can subsequently help lower your BH4, and the NOS gene mutation, you're going to have less BH4. And what's going to happen is if you're drinking, smoking, have high stress, drinking caffeine and other high oxidizing foods, it can cause stress in the mitochondria. And if that happens, you're going to wind up hypersensitizing your T cells, which causes autoimmune issues. When it comes to T cell hypersensitivity, and it's not right away, it's delayed usually. Too much oxidative stress of mitochondria is going to affect your T cells, and you're going to have lower BH4, like I said, if you have the NOS mutation or issues there. If mitochondrial respiration is damaged, it will affect the BH4 production. If mitochondrial respiration is damaged, it also hampers T cells and it cannot properly mature. T cell development, function, and survival are intricately linked to DNA methylation and can impact T cell differentiation, memory formation, and response to pathogens, bacteria, fungal, or cancer. So, dysregulated T cells leads to allergies, lack of ability to fight bacteria, virus, or fungal infections, intracellular and intracellular pathogens, inflammation, and cancer. Oh, mast cells and T cells have a very complex bidirectional relationship, so that means they talk to each other. Mast cells can activate T cells by releasing chemotactic factors, presenting antigens, and inducing adhesion molecule expression. So, mast cells can act as antigen-presenting cells to activate T cells, and this is via the MH class 1 or class 2. Mast cells release cytokines like tumor necrosis factor alpha, which can enhance T cell proliferation and function. Mast cells release exosomes, which are small vesicles containing molecules that can transfer signals to T cells, potentially modulating their function. So if you have a mast cell disorder, you want to get your mast cell disorder under control because it's going to start messing with your other immune cells, and eventually it can cause a T cell hypersensitivity. And this is when you start losing your immune tolerance. T cells can also influence mast cell activity. So T cells can also attract mast cells. T cells can directly induce mast cell degranulation, such as via B chemokines. They release cytokines that are pro-inflammatory that influence mast cell function, such as interleukin 9 and interleukin 2, which can promote TREG activation, and those are your T regulatory cells. Activated T cells can release microparticles that can induce mast cell activation, degranulation, and cytokine release. Nutritional iron deficiency is linked to impaired T cell proliferation and delayed hypersensitivity. So humoral immunity is preserved, but iron deficiency anemia is associated with increased case of cancer. Protein tyrosine kinase. That is in the methylation cycle, particularly the BH4 cycle, and that is where the MTHFR 1298 is at. As you can see in the BH4 cycle, the MTHFR 1298 is there. And tyrosine is metabolized through the BH4 cycle through the MTHFR 1298 to be converted into dopamine. So if mitochondria in the BH4 cycle isn't working properly, it affects the tyrosine conversion to dopamine. BH4 is needed for T cell driven autoimmunity and allergic inflammation. Kynurenine is a molecule that breaks down the essential amino acid tryptophan in the BH4 cycle. Kynurenine, it links the immunosuppressive tumor environment to impaired T cell function, i.e., if it's not working properly, higher chances of cancer cells growing and evading immune detection. Adding more protein to diet can further lead to immune response, confusing your immune system and breaking the immune tolerance you have. The microbiome is going to play a major role in this as well. So as you can see, there's a very close tie between methylation, mitochondria, inflammation, and your T cells. Helper T cells. T helper cells are responsible for coordinating much of the immune response, proliferating helper T cells that develop into effector T cells, meaning it's a group of cells that includes several T cell types that actively respond to a stimulus. This includes CD4+, those are your helper T cells, CD8+, those are your killer T cells, and your T regulatory cells, called TREGs. These effector T cells differentiate into two major subtypes of cells known as Th1 and Th2, also known as type 1 and type 2 T helper cells. So there are two main categories of the T helper cells, that is Th1 and Th2, or type 1 and type 2 T helper cell. However, there are other types of T helper cells, and you can see on the top right corner how I have listed out the rest of them. The TFH is the follicular T helper cells, and those are in the lymph nodes. The Th17 is a subset of the CD4+ T helper cells. So the Th17 is still a helper cell, it's just a lower ranking helper cell than than the CD4 plus T helper cells. So as we learned earlier, an antigen-presenting cell locates the intruder, pathogen or bacteria, and it gobbles it up into the phagosome. And then when it breaks it all up, it starts sending the pieces and parts to the surface of the cell, and it uses the major histocompatibility complex to connect with the T cells. And we learned this a little bit ago. T cells can't recognize evidence of an intruder unless it's attached to the major histocompatibility complex. CD8 cytotoxic or killer T cells can only bind to MHC1. CD4 helper T cells can only bind to MHC2. T helper cells or class 1 helper cells lead to an increased cell-mediated response. They're usually against intracellular bacteria and protozoa. They are triggered by polarizing cytokines interleukin 12 and effector cytokines interferon gamma and interleukin 2. Cytokines are messengers that help the immune cells communicate. Polarizing cytokines and effector cytokines have different roles in shaping the immune system. The main effector cell of the T helper cell class 1 immunity are macrophages, as well as CD8 T cells, which are your killer T cells, your IgG, your B cells, the interferon gamma, and the CD4 T cells, which are your helper T cells. The key transcription factors are STAT4 and TBET. Now, I don't expect you all to know this, but for some of you out there who have gone through extensive testing may understand what this is. Interferon gamma, secreted by CD4 T cells, can activate the macrophages to phagocytose and digest intercellular bacteria and protozoa. And as we learned earlier, the bacteria is engulfed and locked into a structure called a phagosome. And that's basically like a digestive system that is ripping apart the bacteria or foreign object in it. And then when it's done, it takes those pieces and particles of the bacteria and puts it on the cell surface so that the T cells can be triggered. They can also be triggered by cytokines, interleukin 12, and effector cytokines, interferon gamma and interleukin 2. And the thing I have to mention here is, so if you have a mast cell disorder, you're going to wind up triggering these on a regular basis, because remember, your mast cells are constantly degranulating, and one of the things they let out are cytokines. So cytokines, like we have talked about before, are the messengers that help the immune cells communicate. And the polarizing cytokines and effector cytokines have different roles in shaping the immune response. So remember, some suppress the immune and some promote immune and inflammation. The main triggering of the T helper cells class 1 are the macrophages, as well as the CD8 plus T cells, those are your killer or cytotoxic T cells, the IgG, B cells, interferon gamma, and the CD4 T helper cells. Th1 overactivation against autoantigens will cause type 4 delayed hypersensitivities, and we will get into hypersensitivities here in a minute. Or type 1 diabetes belongs to this type of autoimmunity. The Th2 T helper cells lead to a humoral immune response, typically against extracellular parasites. They are triggered by the polarizing cytokines interleukin 2 and interleukin 4. Effector cytokines are interleukin 4, 5, 10, 13, and 25. Now, for many of you, this isn't going to mean a whole lot, but for those of you who have undergone extensive testing and know exactly what interleukins you're throwing too much of, this is triggering your T helper cells, the class 2. The main effector cells are eosinophils, basophils, and mast cells. So if you're having allergic reactions or intolerances, this is going to ramp up your immune system and trigger these helper cells. Interleukin 4 is a positive feedback cytokine for Th2 cell differentiation. It stimulates B cells to produce antibodies, which in turn stimulates mast cells to release histamine, serotonin, and leukotrienes. And this can cause bronchoconstriction, intestinal peristalsis. It's the wave-like movement that moves your food through the intestines, and its opposite is gastroparesis. Also affects gastric fluid acidification to expel helminths, which is a parasitic worm.
And interleukin 5 from the CD4 T helper cells will activate the dendritic cells. And Th2 overactivation against autoantigens will cause type 1 IgE-mediated allergies and hypersensitivity. So allergic rhinitis, atopic dermatitis, and asthma all fall under this category. In addition to expressing different cytokines, Th2 cells differ from Th1 cells in their surface glycans, which makes them less susceptible to some inducers of cell death. Okay, so polarizing versus effector cytokines. As we've been talking about, cytokines are messenger molecules that help the immune cells communicate. Polarizing cytokines and effector cytokines have different roles in shaping the immune system. So polarizing cytokines are like the instructors. They give orders. And effective cytokines are like the action heroes. They actually take action. So let's look at polarizing cytokines for a moment. These cytokines are exactly this. They train or program immune cells, especially T cells, to develop into specialized types. They are also released early in the immune response to tell the T cells what role they should take. They polarize naive T cells, which are untrained, into specific functional types. So Th1 cells fight virus and intercellular bacteria. Th2 cells fight parasites and other trigger allergy responses. Th17 cells are important for inflammation and autoimmune reactions. And Tregs, which are T regulatory cells, help calm down the immune system. So some examples of this are interleukin 12. It tells the T cells to become a Th1, and that attacks the virus and bacteria. Interleukin 4 tells the T cells to become Th2, which defends against parasites and allergens. Tumor growth factor Bravo in interleukin 6 tells T cells to become Th17, which then triggers inflammation. And tumor growth factor Bravo by itself tells T cells to become Tregs. And those are your T regulatory cells, and that's what calms down your immune system and helps keep it from running amok. Now, the effector cytokines, or the action heroes of your immune system, these cytokines are released and activated by immune cells to fight infections. They cause inflammation or regulate the immune response. After T cells have been polarized, they start producing effector cytokines, which actually carry out their immune functions. Think of them as fighters or workers taking action during a battle. Examples of effector cytokines based on T cell type. Th1 is the interferon gamma, then this activates macrophages to kill bacteria and viruses. Tumor necrosis factor alpha increases inflammation to fight infection. With the Th2 cells, interleukin 4, 5, and 13 helps B cells make antibodies and fight parasites and allergens. Th17 cells, the interleukin 17 and 22, causes inflammation and helps fight fungal and bacterial infections. And with T regulatory cells, interleukin 10, tumor growth factor Bravo, suppress inflammation and prevent autoimmunity. So as always, I always have extra slides posted on the slide to help support the information there. So please feel free to pause and read this, or you can find the slides in the Cycle of Illness Immune System slides. So this slide is more supplementary, but you've got the T helper cells, where they mature and how they function and what chemicals they release and the byproduct of what happens when that happens. So Th1, they participate in the mediated immunity. They are essential for controlling such intercellular pathogens like viruses and certain bacteria. They provide cytokines mediated to help cytotoxic T lymphocytes, possibly the body's most potent weapon against intercellular pathogens. They release interleukin 12, interferon gamma, and tumor necrosis factor Bravo. And in turn, this winds up stimulating macrophages to kill bacteria, recruiting other leukocytes to promote inflammation, and they act on B cells to promote antibody class switching. They help cytotoxic T cells do their work and probably help convert some of them to memory cells. Th2 provides help to the B cells and are essential for the production of IgE antibodies, and those are your allergies, and assist in the production of some subclasses of IgG as well. Antibodies are needed to control extracellular pathogens, which, unlike intracellular parasites, are exposed to antibodies in the blood and other body fluids. Th2 releases interleukin 4, 13, and 5, and they are produced when antigen-presenting cells, APCs, present antigens like a parasite or allergens to the T cell receptors for antigens, and paracrine stimulates interleukin 4 and interleukin 2. Now, the TFH, these are called follicular helper T cells. These also provide help to B cells, enabling them to develop into antibody-secreting plasma cells. This occurs in the lymphoid cells. It is called follicles in the lymph nodes. They are the most abundant helper T cells there are, and they also release cytokines. They are found in nests of B cells in the lymph nodes. Binding interactions stimulate B cells to undergo class switching or accept the IgE. They undergo affinity maturation. They form antibody-secreting plasma cells and memory B cells, and it is not clear whether the T follicular helper cells represent a distinct class of T helper cells or are simply a further stage in maturation of the Th1, Th2, or Th17. The Th cells are T cells that haven't fully matured and are awaiting a single signal to tell them what type of T cell you need them to be, or if they're their own distinct class. Th17. These are a subset of CD4 plus T helper cells. They are found between the external environment and the internal environment, such as your skin and lining of the GI tract. They are most likely start out like other naive Th cells, but when exposed to cells presenting antigens, they enter a pathway distinct from Th1, Th2, and follicular T helper cells. They release cytokines, transforming growth factor Bravo, and interleukin 6. They are situated in the lining of the GI tract and cells and are positioned to attack fungi and bacteria at those locations. They do this by secreting defensins and recruiting scavenger cells, especially neutrophils, to the site. The result, clearing away invaders with accompanying inflammation. But inflammation is a double-edged sword, so it's not surprising that Th17 cells have implicated a potent effector such as damaging inflammatory disorders such as Crohn's, which is inflammation of the small intestine, ulcerative colitis, which is inflammation of the large intestine, and psoriasis, which is inflammation of the skin. A balanced immune system requires proper regulation of all of these pathways. Methylation, oxidative stress, BH4, NOS, and immune organs like your lymph nodes and spleen influence how these pathways function. So if you're experiencing pain in all these areas, you need to take that as a giant red flag, and you need to take it seriously and start healing your body by first eliminating the things that are triggering your immune system. Alright, so I don't want to confuse you too much. But as we said, the methylation, oxidative stress, BH4, NOS, and immune organs like the lymph nodes and spleen influence how these pathways function. Now, STAT4 and STAT6 are proteins, act like messengers that respond to cytokines and tell the T cells how to behave. The TBET and GATA3, the immune system's master regulators. These are transcription factors that control how the T cells develop after cytokines activate STAT proteins. The transcription factors are proteins involved in the process of converting or transcribing DNA into RNA polymerase. These initiate and regulate the transcription of genes. And you'll see here STAT4 is activated by interleukin 12. And if it's overactive, it will create autoimmune diseases. And if it's not active enough, weak defense against viruses and bacteria, so you'll be more susceptible. STAT6 and TBET, they are activated by interleukin 4 and STAT4 and T helper cell class 2 development and master regulators. If they're overactive, they cause allergies, asthma, and too much inflammation. If they're not active enough, a weak defense against parasites and poor Th1 response equals poor immunity. The GATA3 is activated by the STAT6. This is the T helper cells class 2, Th2, master regulator. And if they're overreactive, it'll cause allergic diseases. If they are underactive, That means they will have poor Th2 response, which means weak antibody production. If there's not enough antibody production, it's going to make it more difficult for you to clear pathogens, viruses, and other foreign debris that doesn't belong. So the STAT4 is activated by interleukin 12, and it tells naive T cells to become Th1 cells, which are frontline fighters against viruses, bacteria, and intercellular pathogens. This will lead to the production of interferon gamma, a major effector cytokine that supercharges macrophages and your immune response. How it's connected. Th1 cells activate B cells to produce IgG antibodies, and that is good for fighting viruses and bacteria. But an overactive STAT4 leads to chronic inflammation and autoimmune diseases like rheumatoid arthritis. And weak STAT4 leads to poor immune defense, which results in more infections and weak Th1 response. Now, TBET is the Th1 master switch and the immune system's general, so to speak. It is activated by STAT4 and interleukin 12. This tells the Th1 cells to be aggressive defenders against virus and bacteria. And this leads to increased interferon gamma and supercharges macrophages, and it suppresses Th2 and Th17 cells, which prevents unnecessary inflammation. And how it's connected is without TBET, Th1 responses fall, which results in a higher risk of viral and bacterial infections. An overactive TBET results in autoimmune diseases like rheumatoid arthritis, MS, etc., due to excessive Th1 activation. And if you have mast cells, methylation, mitochondrial damage, and a weak microbiome, this is so going to affect you, and you're going to wind up having chronic immune activation. Alright, here is a chart of the interleukins and what each one of them does and what they trigger and what can be caused from it. I'm not going to read over each and every single one of these. It'll take forever. So please pause the video to take a look at it, or you can find this in the Cycle of Illness Immune System slides. There is a lot of good information in these two slides that can help you to understand what is happening in your body and why you're having aberrant cell responses. This can actually help explain some of that. In the slideshow, I also have the CD charts. For the complete CD charts with their information, please see the following links below. There is way too much information in each chart to include in this paper or this video. The web addresses are subject to go down and not come back up randomly. This was a current list of CD charts at the time that I wrote this. Personally, I like the HCDM website. That seems to be the best one. And I'm going to tell you right now, these types of sites pop up and go down like you wouldn't believe. So hopefully they are still up and working by the time you see this. I also have all the TH pathways mapped out. This might be a little bit difficult to read on the computer screen or TV, but these are actually in the slideshow as well, and it can show you all the pathways and all the chemicals that come from it and what they trigger and how it all flows. Killer T cells. These are the CD8+, or the cytotoxic T cells. Killer T cells are known as cytotoxic T lymphocytes. They are a type of white blood cell that plays a crucial role in the immune system. They are also part of the adaptive immune system. They identify and eliminate infected cells, cancer cells, virus-infected cells, and bacteria-infected cells. Killer T cells have a CD8 receptor, and that's why they're called CD8+. And the helper cells have the CD4 receptor. And you can see on the B cells versus T cells diagram with all the receptors on there exactly what I'm talking about. I also place this in the B section. When CD8 receptors interact with the major histocompatibility class 1, when healthy cells are infected. When the CD8 receptor recognizes an infected cell, it activates the killer T cells to destroy the infection. And the CD8 receptor interacts with cells called the major histocompatibility complex, the MHC1, as we've been discussing this whole time. Helper T cells help activate killer T cells when they release cytokines. And cytotoxic T cells, or killer T cells, form in the bone marrow, then move to your thymus to mature.
Your thymus is much larger when you're a child and gets smaller as you age. Approximately age 20, your thymus releases significantly less T cells. Your diverse supply of T cell depends on your body's ability to make copies of T cells you already have. And the highest concentration of T cells are in the lymphatic system, in your lymph nodes, your bone marrow, intestines, lungs, spleen, and tonsils. This is just like that time when you were a kid in school and the teacher would stomp their foot to let you know that the information that you're receiving at the moment is on the test. Well, I am giving you the same proverbial metaphorical foot stomp, except I'm going to say that when these areas of your body are in a lot of pain, you need to take this seriously. Get rid of the triggers that are causing this. The normal range of T cells, generally a CD4 count helper T cells that are between 500 and 1200 cells, and a CD count of cytotoxic T cells between 150 and 1000 cells per cubic millimeter is considered normal. Low T cell counts can signal a range of issues. A scan can be a side effect of certain treatments or medications. Certain conditions you require, like HIV, can cause low T cells. Not having enough T cells can make it harder for your body to protect you, and too few T cells can be life-threatening. High T cell count means your body is ramped up to fight infections and can be a sign of certain cancers. Conditions and disorders of the T cells. Autoimmune diseases affect your T cells. Acute lymphocytic leukemia starts in the blood and bone marrow. Hodgkin lymphoma starts in the lymph system. HIV, Job syndrome, is a rare immune disorder that causes repeat infections. Thymic asplasia, people born with an underdeveloped thymus. Severe combined immunodeficiency, SCID, is a rare genetic disorder that involves a weakened immune system resulting from the problems with T cells and other lymphocytes. Wiskoff-Aldrich syndrome is a rare genetic condition that involves immune system issues. It includes atypical white blood cells. DeGeorge syndrome is a genetic disorder that can prevent the body from making T cells, and most of this is caught very early on in your life, with the exception of the acute lymphocytic leukemia or the Hodgkin's lymphoma. Those can be caused if you're not taking care of your health and eliminating the triggers, especially after you start experiencing pain in all those key organs I was talking about. Alright, so how to heal it and keep it healthy. You need to avoid alcohol or consume it in moderation only. Now, if you already have MTHFR, fatty liver issues, do not drink alcohol, and for all the love in the world, do not smoke. Make sure you eat a diet filled with fruits, vegetables, whole grains, and lean proteins and healthy fats. And I know for some of you this is very difficult to do because of our methylation impairments. Please look at the methylation videos and slides so you can understand what your body can and cannot handle and work within the blueprint that you were given. You also want to exercise consistently, incorporating strengthening, flexibility, and aerobic activities. You also want to sleep 7 to 8 hours a night, wash your hands frequently with soap and water to avoid germs, and wear a mask when necessary if you are susceptible to significant allergies, triggers, or immune weaknesses. Now, just a quick note on the side, what is the difference between killer T cells and natural killer cells? Well, killer T cells attack viruses when activated by an antigen, so the adaptive immunity. And natural killer cells don't need activation to attack harmful cells, and that is part of your innate immunity. And we'll be talking about that next. Hang in there, guys, we're almost through. But before that, it's time for How Is It All Connected? Alright, T cells and the BH4 of your methylation. BH4 is a critical molecule that helps immune cells, including T cells, function properly. So, if you have the MTHFR 1298, you have to monitor this closely. So you're going to want to watch your stress levels because this will crash this very quickly. You're also going to want to make sure you're watching what you eat. You want to keep out as much oxalates as possible because you don't want to cause more damage to the body and cause more oxidation to the body. The other thing you also have to do is take out your triggers, because every time you continue eating your triggers, you're just keeping your immune system and your nervous system stuck in fight or flight. And I know this can be a very tricky spot to be in. So if you're running with neurological issues, it's already telling you have low BH4. And if you have the NOS gene mutation in methylation as well, this will also contribute to this. And we'll talk about that in a minute. So your T cells need BH4 to grow, activate, and fight infections effectively. And without enough BH4, the T cells can become weaker, slower, and less effective in their immune response. Now, I was talking about the NOS. The T cells in NOS, in methylation, is an enzyme that produces nitric oxide, which helps the immune cells kill invaders. T cells use nitric oxide to regulate inflammation and fight infections, but too much nitric oxide can cause excessive inflammation and damage healthy cells. BH4 helps NOS to work properly, so it's a trickle-down effect. So you must make sure that nitric oxide production is balanced. All right, T cells and your lymph nodes. Your lymph nodes are training camps for T cells. This is where T cells mature, get activated, and prepare to fight infections. And remember, they are also trained in the thymus. If an infection is detected, T cells multiply inside the lymph nodes before heading to the bloodstream to attack. So swollen lymph nodes happen when T cells and other immune cells are working overtime to fight infections. T cells and your spleen. The spleen is a filter for blood and a backup immune system hub for T cells. T cells gather in the spleen to detect threats in the blood and help fight infections. If the spleen isn't working properly, T cells may not be as effective in recognizing and fighting infections. T cells and oxidative stress in mitochondria. Mitochondria are the powerhouse of the cells, and this is what gives you your energy, including T cells. When T cells are activated, they need a lot of energy to multiply and fight infections. Oxidative stress happens when mitochondria produce too many harmful molecules, which are called free radicals, which can damage T cells and make them less effective. So if you are drinking caffeine, if you are smoking, this all contributes to oxidative stress. And a lot of the junk food that's in the modern Western diet is also oxidative. Chronic oxidative stress weakens T cells, making them more prone to dysfunction and increasing inflammation in the body. And another point I want to make here, stress. Stress also greatly ups your oxidative stress in your body and in your mitochondria. You'd be surprised what stress can do. So BH4 helps the T cells function properly. NOS helps T cells regulate inflammation with nitric oxide. And lymph nodes and spleen are hubs where T cells activate and prepare to fight infections. And mitochondria give T cells energy. But too much oxidative stress greatly weakens them. Now, if your BH4 is low, your NOS is imbalanced, or oxidative stress is too high, T cells can become dysfunctional, leading to weaker immunity, more inflammation, and a higher risk of chronic illness. BH4 and NOS regulate T cells and B cells, ensuring they work properly and don't overreact. T cells activate B cells in the lymph nodes and spleen, helping them create antibodies. If oxidative stress is too high, mitochondria don't produce enough energy, and then the B cells can't fight infections properly. And if NOS isn't balanced, B cells might get confused and start attacking healthy cells, which is an autoimmune reaction. And without enough BH4, both B cells and T cells become sluggish, leading to weaker immunity and chronic illness. So balanced BH4 and NOS, healthy T and B cells, strong immunity. Too much oxidative stress, and this includes what you're eating and your stress levels, so stop drinking and stop smoking if you are. And they are weakened or overactive B cells, leading to immune dysfunction. And dysfunctional lymph nodes and spleen equals poor B cell activation, leading to chronic infections or autoimmune disease. This is a very delicate balance, is what keeps your immune system strong but not overreactive. When one part of the cycle is off, B cells, T cells, and overall immunity can definitely suffer.
guys, we're almost done. Natural killer cells. Natural killer cells are white blood cells that destroy infected and diseased cells. They can destroy harmful cells in the early stages, preventing virus and cancer cells from spreading. They are part of the innate immune system, so they don't need to be triggered. They can do it on their own. Your innate immune system is your body's first line of defense against all threats. This includes physical barriers such as your skin, mucus, and membranes. One of the earliest cell types to arrive at target organs of inflammation. Its role can actually be a little bit paradoxical. Additional elements such as organ-specific factors and cellular interactions. So this means that it may influence the divergent functions of natural killer cells in different organs during different pathological conditions. These cells can recirculate between organs, and chemokines produced by cells that are unique to a specific organ may have a role in orchestrating natural killer cell migration to each organ. Natural killer cells can be divided into functionality distinct subsets. The main two we will be talking about is CD16 and CD56. So, CD56, bright. These are natural killer cells. These natural killer cells express high levels of CD56 and are often found in tissues, particularly secondary lymphoid tissues. They are generally considered less mature and have a greater propensity for cytokine production, and that means it plays a role in immunoregulation. They respond strongly to soluble factors like cytokines and are potent producers of cytokines such as interferon gamma and tumor necrosis factor alpha, which exert immunoregulatory effects. Now, CD56-DIM is a type of natural killer cells, and these natural killer cells express low levels of CD56 and are the majority of natural killer cells found in peripheral blood. They are considered more mature and have a greater cytotoxic ability. The expression of CD56 can be used as a phenotypic activation marker, which means that upon activation, both CD56 bright and CD56 dim natural killer cells can upregulate their CD56 expression. So kind of think of it as like turning up the volume on those particular cells and making them upregulate their cytokine production, which can lead to a role in immunoregulation. So it can mess up your immune system. And natural killer cells exposed to an immunosuppressive environment may downregulate their CD56 expression. CD16 is also known as the Fc receptor 3. Now remember, we talked about the Fc receptors earlier in the video. The Fc receptors are a group of proteins found on the surface of many immune cells, and they bind to the Fc region of antibodies on other cells. And that's what tells the immune system that you have an infected or damaged cell. And don't forget, we also talked about if there's mutated Fc receptors, it's going to completely change how your immune system responds. CD16, or the Fc gamma receptor number 3, facilitates antibody-dependent cellular cytotoxicity. This binds to the Fc portion of antibodies, triggering the natural killer cell's activation and leading to a target cell lysis and cytokine production. So basically, if you look at the top right-hand corner here, the diagram we have here, we have an infected cell. The natural killer cell binds to the protein that the infected cell is giving off. And then what happens is the natural killer cell sends all of its cytokines into the viral infected cell to destroy it. So you can see how the CD16, or also known as the Fc gamma receptor number 3, if that's not working properly, it's going to change how your immune system and your natural killer cells respond, and this can cause a lot of problems. So the low affinity receptor for IgG, and when antibody-coated large cells like a virus infected or cancer cells are recognized, this triggers cytotoxic activity in cytokine production, which is a pro-inflammatory cytokine, and chemokines, both of which contributes to tumor killing and therapeutic efficacy. It also triggers phagocytosis, and CD16 downregulation can represent a way to moderate natural killer cell responses and maintain immune homeostasis. The Fc receptor CD16 is present on essentially all CD56 dim peripheral blood natural killer cells, which targets through direct killing and cytokine production. It recognizes and kills cells that are missing the major histocompatibility class 1 molecules, or cells that express stress-induced ligands on their surface. They also release cytotoxic granules, which are perforin and granzymes that induce apoptosis, which is programmed cell death for the infected or damaged cell in the target cell. So in the top right-hand corner picture there, where you've got the natural killer cell and the viral infected cell, if you look closely, you'll see that you have a receptor there. There's the red one, and then there's the blue one. But if you look at the other two options, you'll see the major histocompatibility complex is much bigger and much longer. Whereas in example number 3, it's extremely short, and the natural killer cell doesn't make contact with the major histocompatibility complex. So this is like a backup system to your T cells, B cells, etc., and it kills the viral infected cell, all because it can't make contact or join up through the major histocompatibility class 1. Kind of think about it as like trying to make a call. You're trying to make a connection, but you can never connect. And because you can never connect to the person you're trying to call, that's when it clues you in that there might be something wrong. And that is when the natural killer cells act and kill the infected cell. And remember, T cells absolutely need the major histocompatibility complex in order to be triggered and kill the pathogen virus within that cell. And so the natural killer cell is like a backup, so anything the T cells can't connect to, the natural killer cell will do it. In a nutshell, that's the simplest explanation. This can kill cells without prior activation or antigen recognition. Killer T cells kill cells after activation by a specific antigen and are part of the adaptive immune system. They can kill a variety of cells that are perceived as threats. They have rapid response time, but they do not need previous exposure to a pathogen before they can destroy it, unlike T cells. A natural killer cell's primary job is to kill. However, these cells also communicate. They release proteins called cytokines, and they direct other cells in your immune system to attack harmful cells and pathogens. They live in the body in various locations. Your bone marrow, the peripheral, which is your blood that circulates through the body outside of the heart and lungs, your secondary lymphoid organs, which are your lymph nodes, spleen, tonsils, and thymus, liver, and lungs, as well as in the uterus, and this is a major subset in the pregnant uterus. It's a unique phenotype resembling an early developmental state, and this plays a crucial part in mediating uterine vascular adaptations to pregnancy, and it promotes the maintenance of healthy pregnancies. It's also in the skin and gut, and they are detected in healthy lymphoid tissues of the lung, skin, and gut. They are recruited to these tissues during infection or inflammation. This is important to know if you have digestive issues like IBS, IBD, Crohn's, ulcerative colitis, etc., because they likely to have crucial roles in controlling the inflammatory response in those areas. In mouse studies with multiple sclerosis, they have shown that natural killer cells arrive in the central nervous system before the T cells. They have a protective role in the development of the central nervous system inflammation, and they are most likely by killing the central nervous system resident microglia that activates the effector T cells. It's also, the natural killer cells are also crucial for the protection against antibody-induced arthritis. It makes up of about 5 to 10% of lymphocytes circulating in your blood. They have an approximately a 2-week lifespan, and at any given time, adults have more than 2 billion natural killer cells. They activate when signals that prevent them from attacking, like the major histocompatibility class 1 molecules, are absent or downgraded on a target cell. So diseases of the natural killer cells are autoimmune diseases. Problems with the natural killer cell's function may play a role in autoimmune diseases. Asthma, It has a role in the body's inflammatory response with asthma, and they can increase or decrease inflammation depending on various factors. Inflammatory bowel disease. It also plays a role in your body's inflammatory response to bacterial infections in your gut. So you can see where cell damage, or if another part of your immune system is not working properly, this has a massive cascade effect. So it's extremely important to make sure you take care of your body so that your immune system stays in homeostasis. The other thing I wanted to mention was that this is why I also promote fasting. So these cells can live about 2 weeks. And what's happening is if you have an overproduction of these cells and they're over-responding, they're kind of keeping everything going in a stress-induced overactive immune system in your body. And by fasting, you're shutting down all these processes, and any of these immune cells that have become aberrant, it shuts them down. Some of them you have to wait till they die off in order to begin the healing journey. So there are 2 conditions that affect natural killer cells. The 2 immunodeficiency conditions are specific to natural killer cells, and both result from genetic mutations at birth. So this is something that will get caught early in your life. There's the classical natural killer cell deficiency, and this involves having no or very few natural killer cells in your blood. And to have classical NKD, less than 1% of your circulating lymphocytes are natural killer cells. The other one is functional natural killer cell deficiency, and this involves having natural killer cells that do not function as effectively as normal natural killer cells. Both conditions make you more susceptible to infections: HPV, herpes, cytomegalovirus, Epstein-Barr virus, and varicella-zoster virus, and this is the one that causes chickenpox. It's that time again for How Is It All Connected? Natural killer cells are very highly connected. They are a critical part of the innate immune system, and unlike B and T cells, which need activation and training, natural killer cells act as fast and are the body's first line of defense against viruses, cancer, and infected cells. Now, natural killer cells and B cells, the antibody connection. Natural killer cells rely on B cells to produce antibodies that help them recognize and kill infected or cancerous cells. B cells, through IgG antibodies, trigger antibody-dependent cellular toxicity, where natural killer cells attack antibody-coated cells. If B cells are dysfunctional, natural killer cells have less guidance in targeting threats and reducing immune efficiency. Now, balanced natural killer and B cell connection equals a strong antibody production, and that equals cells get better targeting signals to kill infected or cancerous cells. Now, dysfunctional B cells will equal weak antibody response, which results in natural killer cells struggling to find and destroy threats. Natural killer cells and T cells, the immune coordination. Helper T cells, those are your CD4, help activate natural killer cells by producing interleukin 2 and interferon gamma, which boosts their killing power. Cytotoxic T cells, those are your killer T cells, and natural killer cells work together to destroy virus-infected cells and cancer cells. If T cells fail, natural killer cells have to work harder, leading to immune exhaustion and weakened defense. So balanced T cell and natural killer cell connection, T cells boost natural killer function, ensuring effective immune responses. Weak T cells, the natural killer cells, become overworked, and then this leads to immune fatigue and increased infections. Natural killer cells and BH4. Remember, BH4 is in your methylation, and this is the energy and defense regulator. BH4 is crucial for natural killer cell activation and helps them produce cytotoxic molecules like perforin and granzymes. Low BH4 levels make natural killer cells sluggish, reducing their ability to kill infected cells. BH4 also protects natural killer cells from oxidative stress, keeping them functional. So balanced BH4, natural killer cells remain strong and responsive. Low BH4, natural killer cells will become weak, making infections and cancer far more likely. So it's really important that you're balancing your methylation cycles, because the BH4, if you run out of it, BH4 affects your moods, your energy, and also your ability to detox. And if it's not working properly, it will give you a host of neurological conditions. So it's extremely important for you to keep your methylation as healthy as possible, because it has a direct link to how your immune cells work. Natural killer cells and NOS. This is where inflammation control comes in. Nitric oxide synthase, that's the NOS. This is also part of methylation, produces nitric oxide, which helps natural killer cells kill infected and cancerous cells. Too much nitric oxide and the natural killers will go into overdrive, causing inflammation and autoimmunity. Too little nitric oxide and the natural killer cells become weak, allowing infections and cancer to grow. Balanced NOS equals natural killer cells killing infections without excessive inflammation. And if there's a NOS imbalance, too much equals autoimmune risk and too little weak immune response. Natural killer cells and the lymph nodes, and this is your immune communication. Natural killer cells patrol lymph nodes looking for infected or cancerous cells. And if an infection is detected, natural killer cells send signals to B and T cells, boosting immune activation. If lymph nodes are dysfunctional, the natural killer cells don't get proper signals, leading to immune failure. So healthy lymph nodes, the natural killer cells quickly detect and respond to threats. Weak lymph nodes and your natural killer cells lose key activation signals, which weakens your defense. Natural killer cells and the spleen, the blood patrol. The spleen is a major hub where natural killer cells detect and kill cancerous and infected cells in the bloodstream. If the spleen isn't functioning properly, natural killer cells can't patrol effectively, increasing infection and cancer risk. So a healthy spleen, the natural killer cells efficiently patrol for infection and cancer cells. A dysfunctional or damaged spleen equals weak surveillance, which results in more infections and disease progression. Natural killer cells and oxidative stress in mitochondria. This is the energy factory. Natural killer cells need a lot of energy from mitochondria to track down and kill infected cells. Oxidative stress from damaged mitochondria reduces natural killer cell function. If oxidative stress is too high, the natural killer cells become exhausted and cannot function. If oxidative stress is too low, natural killer cells don't get enough energy to fight infections. So balanced mitochondria and oxidative stress results in the natural killer cells remaining powerful and responsive. But excessive oxidative stress will result in the natural killer cells becoming dysfunctional and increasing your disease risk. So you might notice that if you have an autoimmune condition and you kind of fall off the wagon with your food and your habits, etc., you'll notice a definite progression in your autoimmune condition. And this is part of that progression. So, natural killer cells are the body's frontline defenders against viruses, infected cells, and cancer. They depend on B cells, T cells, BH4, NOS, and lymph nodes, spleen, and mitochondria to function properly. When everything is balanced, the natural killer cells protect against disease. When the system is off, natural killer cells can either weaken, which equals increased infections and cancer, or become overreactive, which results in autoimmunity and inflammation. This is how natural killer cells fit into the immune cycle and interact with the B cells, T cells, and other immune factors. So to recap, natural killer cells work with B cells, and B cells make antibodies that help the natural killer cells target infections and cancer. Natural killer cells work with T cells. T cells boost natural killer activity with interleukin 2 and interferon gamma. BH4 fuels natural killer cells and ensures that they stay strong and effective. And NOS regulates natural killer cell activity, preventing immune exhaustion or autoimmune reactions. Lymph nodes help train natural killer cells, ensuring they detect and respond to threats. And the spleen helps natural killer cells patrol the blood, killing infections and cancerous cells. Oxidative stress and mitochondria control natural killer cell energy, affecting their ability to fight. So balance is the key. We have got to stay in the Goldilocks zone. All right, so the NOS in the BH4 is part of methylation, and the methylation plays a critical role in regulating natural killer cell function, including their activation, energy production, and ability to kill infected or cancerous cells, and inflammation control. If DNA methylation is too high or too low, natural killer cells can become dysfunctional, leading to weakened immunity or excessive immune activation. DNA methylation is like an on-off switch for genes, determining which genes are activated or silenced. If it does affect natural killer cells by controlling their ability to recognize, attack, and eliminate threats. Methylation controls natural killer cell activation and function. Proper methylation helps natural killer cells recognize and kill infected cells, tumors, and viruses. Helps them produce cytotoxic molecules like perforin and granzymes that destroy the harmful cells, and the balance immune responses so they don't attack healthy tissues. If methylation is off balance, if you're an over-methylator, natural killer cells become suppressed, leading to weaker immunity and higher cancer risk. If you're an under-methylator, and mast cell patients generally are under-methylators, anyone who has histamine intolerances are generally an under-methylator, then NK cells become overactive, increasing risk for autoimmune diseases. Methylation regulates cytotoxicity, so the natural kill cells' killing power. Natural killer cells destroy infected and cancer cells by releasing perforin. The perforin punches holes in the enemy's cells. Granzymes enter the enemy's cells and triggers cell death. Methylation controls the genes that regulate perforin and granzymes. If these genes are over-methylated, natural killer cells lose their ability to kill cancer and infected cells. If these genes are under-methylated, natural killer cells may over-attack, contributing to inflammation and tissue damage. So balanced methylation equals natural killer cells are able to maintain strong cytotoxicity without excessive inflammation. Methylation and the BH4, which is the natural killer cell's energy supply, is essential for natural killer cell activation and energy production. Methylation controls BH4 levels by regulating the MTHFR gene, which affects folate and SAMe metabolism. If methylation is dysfunctional, low BH4, the natural killer cells lose energy and become weak and ineffective. Excessive BH4, overactive natural killer cells cause inflammation and oxidative stress. So, as we were saying, balanced methylation equals natural killer cells having the right amount of energy and the right chemicals to perform properly. Alright, now that we have finished all of the immune system cells, we need to look at hypersensitivity reactions. There are four main types of hypersensitivity. Type 1 is fast and allergic, and that is usually IgE and histamine. Type 2 is antibody attack on cells, which is IgG for infections and IgM. Type 3 is immune complexes get stuck, and this is again IgG, and this deals with infection and inflammation. And type 4 is delayed T cells attack. So there are no antibodies, just inflammation. So type 1 is an immediate hypersensitivity and is most commonly known as allergic reactions. The immune system overreacts to harmless substances like allergens, pollen, peanuts, and pet dander. The key players are IgE antibodies and mast cells, which release histamine, cytokines, etc. The timing is extremely fast, and it happens within minutes of exposure. Examples are seasonal allergies like hay fever, asthma attacks triggered by an allergen, anaphylaxis, which is a severe allergic reaction, and pollens, food, drugs, insect stings, etc. Think of it as a hypersensitivity alarm system that freaks out over a false fire alarm and releases histamine, causing swelling, itching, and breathing problems.
Type 2 hypersensitivity reactions are antibody-mediated attack on cells. The immune system mistakenly attacks healthy cells, thinking that they are foreign. The key players are IgG and IgM antibodies, plus complement proteins that destroy cells. And the complement proteins, that is your complement system kicking in. The timing is hours to days after the exposure. And for mast cell patients and those with chronic allergies, the severe ones, this is why you have to wait approximately 3 days to find out what you reacted to, because there is a slower response for certain aspects of the allergens or the immune response. So examples are hemolytic anemia, and this is the immune system destroys red blood cells. Blood transfusion reactions. Graves' disease, and this is where the antibodies attack the thyroid. And rheumatoid arthritis and lupus are part of the type 2 cytotoxic hypersensitivities. Type 3, immune complex mediated hypersensitivity. Immune complexes, antigen plus antibody clumps, build up in tissues and cause inflammation. Key players are the IgG antibodies, immune complexes, and complement proteins. The timing is 1 to 3 weeks after exposure. Examples of this are lupus, SLE, which is the immune complexes damage multiple organs, rheumatoid arthritis, inflammation in the joints, and serum sickness, a reaction to medications like penicillin. Think of it as like a clogged immune pipe. The immune system forms junk clumps that get stuck in the tissues, causing inflammation and damage. And the final is the type 4 delayed hypersensitivity, T-cell mediated. T cells overreact and attack healthy tissues, causing inflammation. The key players are your T cells, macrophages, and cytokines, but no antibodies are always involved. The timing is delayed. It takes 24 to 72 hours to develop. And again, this is another reason why I tell people to fast. It helps shut down these hypersensitivity reactions. Examples to this are poison ivy, contact dermatitis. So if you touch something and you get an allergic response, say like to pet dander, that's what it is. Tuberculosis skin test, and celiac disease, and that is the immune reaction to gluten. Think of it as a slow, smoldering fire. Instead of an immediate reaction, T cells gradually attack, causing delayed inflammation and tissue damage. Now, there are different types of antibodies, the IgG, IgA, IgM, etc. We did cover this briefly earlier, but we'll take a little bit more look at it. The antibodies are also called immunoglobulins, Ig, and they are proteins made by the B cells to fight infections. Each type has a unique role in protecting the body. IgG is the long-term defender. It is the most abundant antibody in your body, and it encompasses approximately 80% of all the antibodies in your body. Their function is to provide long-term immunity after infection or vaccination. It crosses the placenta to protect newborns, and it activates complement. That is a system that helps destroy bacteria. And when we say that it helps activate complement, that means it's activating your complement system. IgG antibodies can be found in the blood, the lymphatic system, and tissues. Examples are it protects against viruses and bacteria like the flu, COVID-19, etc. It also uses monoclonal antibody therapies to help with it. Think of it as an experienced warrior that remembers past infections and is always ready for battle. Now, there's the IgA, the mucosal shield. Its function is to protect mucosal surfaces like the eyes, nose, mouth, lungs, and intestines. It prevents pathogens from sticking to the cells. It's also passed through breast milk to protect infants. It is found in saliva, tears, mucus, breast milk, and the gut lining. So examples of this are it defends against respiratory and digestive infections. It also helps prevent food allergies and gut infections. So think of it as the bodyguard of all the entry points, stopping invaders before they get inside. And obviously if it's not working properly, because it's supposed to help prevent food allergies and gut infections, there would suggest that you're not making enough. IgM, the first responder. This is the largest antibody, pentameter shape, and is the first to respond to infections. Its function is the first line of defense before IgG takes over. It activates the complement system to kill bacteria. It also helps with early immune responses. And it's found in the blood and lymph nodes, but it's too big to enter tissues easily. Examples of this are used in early infection testing, like early COVID-19 antibody tests. It also fights new bacterial and viral infections. Think of it as an emergency first responder that rushes to the fight infections before long-term immunity kicks in. So it works really closely with the complement system. IgE, and this is the stuff that allergies are made of, but it is also the parasite fighter. It is the least abundant but most powerful in allergic reactions. Its function binds to mast cells and basophils, triggering a histamine release. It protects against parasites like worms, ticks, etc. And it causes allergy symptoms like sneezing, hives, asthma, etc. It is found in the skin, lungs, and mucosal surfaces. It is responsible for allergic reactions, and it helps fight parasitic infections. Think of it as an overreactive alarm system. Great for fighting parasites, but can cause allergies when it mistakes harmless things for threats. IgD, the mystery antibody. Alright, well, IgD is the least understood antibody and is less than 1% in the blood. Its function is to help B cells mature and become activated. It may help with immune system regulation. It is found in small amounts in the blood and B cell surfaces. This is still being studied. It is likely important for B cell memory and activation. Think of it as the mysterious backstage manager that helps train B cells but doesn't directly fight in any infections. Alright, we have now covered all the cells of the immune system, hypersensitivity reactions, antibodies, growth factors, interleukins, CDs, etc. So now I'm going to show you how everything is all put together. I've been doing that in short little sections, but now I'm going to put the whole giant picture together. It's the last time for How Is It All Connected? Now, why did I teach you all of this? I am about to wrap it up in a neat little bow. All the videos are going to come and join in this one tiny spot. The whole culmination of all the videos that I've been doing are about to converge in one spot. And you're about to see how everything is so closely integrated together. I bet you were wishing I just started off with this to begin with and skipped all the science in between. But then the knowledge wouldn't be complete and it wouldn't help you as effectively on your healing journey how this all fits together. This whole thing is one big giant puzzle, and we're trying to break it down so that you can see how the immune system's grand design and how it affects everything else. A healthy immune system needs balance. Overactive cells cause autoimmune diseases and allergies, while underactive cells lead to chronic infections and weak immunity. Now, we've talked about the immune cells, their main jobs, and what they're connected to, and what dysfunction does. So, macrophages and dendritic cells detect threats and activate T cells in the lymph nodes. T cells decide what immune response is needed, Th1, 2, 17, or Tregs. B cells make antibodies, IgG, IgE, etc., and they are influenced by the T cells and dendritic cells. Mast cells react to antibodies, releasing histamine and inflammation. The microbiome trains all of these immune cells and influences inflammation levels. Energy and inflammation balance. BH4 and NOS regulate immune activation. Too much equals inflammation. Too little equals weak immunity. Oxidative stress damages mitochondria, affecting all the immune cells and your methylation and your inflammation. Methylation controls immune gene expression, which influences B cells, T cells, macrophages, and even your mast cells. The core players are the B cells, T cells, and natural killer cells. The B cells are antibody factories. They produce antibodies to fight infections. They work closely with the T cells for activation.
They are heavily influenced by the microbiome, which helps train B cells to recognize threats. T cells are the immune generals and assassins. The helper T cells, or the CD4, activates and directs B cells and other immune cells. Cytotoxic T cells, which are your killer T cells, or CD8, destroy infected and cancerous cells. Remember, they are also trained by the B cells and via the microbiome. T cells rely on BH4 and NOS for proper activation and function. Natural killer cells are the innate defense and cancer hunters. Natural killer cells attack and kill virus-infected and cancerous cells without needing prior exposure. They depend on mitochondria and BH4 for energy and on NOS for inflammatory signaling. Additional immune players are the macrophages, dendritic cells, and mast cells. Macrophages are the big eaters and signalers. They engulf pathogens and dead cells through phagocytosis. They present antigens to T cells to kickstart the adaptive immune response. They regulate inflammation through cytokine release. Their function is tied to NOS, BH4, and mitochondrial health. Dendritic cells are the master messengers. They capture and process antigens, then present them to T cells in the lymph nodes. They decide whether to trigger an immune attack or tolerance. Their activity is influenced by the microbiome as well as BH4, NOS, and oxidative stress. Mast cells are the alarm bells. It's kind of like the coal miner's canary. They are also the master switch to the immune system. They reside in the tissues of potential entry points. They release histamine and cytokines to initiate inflammation, especially in allergic responses. They are activated by IgE from B cells and influenced by NOS, BH4, and the microbiome. Overactivation can lead to allergies and chronic inflammation that leads to a vicious cycle, as illustrated in the cycle of illness and as I've been demonstrating throughout this series. BH4, NOS, and their immune connection. BH4 is the immune fuel. They are required for T cell activation, B cell function, and natural killer cell efficiency. It regulates NOS, which is the nitric oxide synthase, and controls immune cell energy production. And remember, to kill off infected cells, they need nitric oxide to do that. Nitric oxide is the nitric oxide synthase. It's also the NOS gene in methylation. This is the inflammation modulator. It produces nitric oxide, which helps immune cells to kill infections. If NOS is too high, it causes chronic inflammation and oxidative stress. So if you have mutations in the NOS gene, this will greatly contribute to that. If NOS is too low, immune cells become weak and ineffective. BH4 and NOS balance. So with all this, you want to see the methylation slides and video to truly understand it, but too much nitric oxide, immune diseases and inflammation. Too little nitric oxide, and you have weak immunity and more infections. Lymph nodes and spleen. The lymph nodes are the training camps where B cells and T cells mature and activate. The spleen is a blood filter that removes infections and trains marginal zone B cells. This acts as a first responder to blood-borne pathogens and works together with lymph nodes to ensure immune cells receive proper signals. Both organs help T cell and B cells interact, learn, and launch immune attacks. It coordinates between them. If either organ is dysfunctional, immune cells don't get proper instruction and leading to autoimmunity or weak defenses. So if you are noticing a lot of pain in your lymph nodes and spleen, please fast and please take this seriously and cut out all your triggers, cut out everything that is harming you. Oxidative stress and mitochondria, the powerhouse and damage control. Mitochondria equals the energy factories for all immune cells. Oxidative stress equals too many free radicals, leading to cellular damage and immune dysfunction. If oxidative stress is too high, immune cells become overactive, causing autoimmunity. If oxidative stress is too low, immune cells become weak and infections can spread. BH4 helps the mitochondria stay healthy by reducing oxidative stress. Stress can crash this whole system. And if you're smoking, drinking caffeine, and drinking sodas, that is causing a lot of oxidative stress to your body. So too much oxidative stress, mitochondrial damage, autoimmunity, and immune cell exhaustion. Too little oxidative stress equals poor immune activation and infections. The methylome, the genetic switchboard. Now, the methylome is in your microbiome. It's a specific section that governs it. The methylome sets a genetic switchboard that controls which genes are turned on and off in B cells, T cells, and natural killer cells. Proper DNA methylation equals healthy immune responses. Too much methylation, immune suppression, and weak immune responses. Too little methylation, and you have an overactive immune response and autoimmune diseases. Methylation and immune balance. It is influenced by factors such as the balance of BH4, NOS, and oxidative stress. BH4 is needed for proper DNA methylation. NOS affects methylation by regulating inflammation, and oxidative stress damage methylation patterns. So inflammation also hampers your BH4, your NOS, everything. So everything is a domino effect, and everything is interconnected. I cannot possibly show you all of the intricate connections that take place in your body. There is just way too many. But I'm hoping through these videos and these slides that you're really starting to see how it's all interconnected. And I'm doing my absolute best to try to illustrate that. The microbiome is the immune system's trainer. The gut microbiome educates immune cells, such as B cells, T cells, natural killer cells, on how to react to threats. A healthy microbiome produces short-chain fatty acids that regulate and promote immune balance. A damaged microbiome leads to increased inflammation, which results in overactivation of T and B cells, and that results in autoimmunity. A damaged microbiome can also lead to weakened immunity, which results in poor B and T cell activation and more infections. The microbiome also affects BH4 levels, NOS activity, and the overall oxidative stress in your body, which affects mitochondria. The microbiome and immune balance. Healthy microbiome equals proper immune function and reduced oxidative stress. A dysbiotic or unhealthy microbiome equals immune dysfunction, inflammation, and chronic illness. So the microbiome trains B cells and T cells in the lymph nodes and spleen. And don't forget, the T cells are also trained in the thymus. BH4 and NOS regulate immune activation and inflammation. Mitochondria provides the energy for immune cells, but oxidative stress must be controlled. The methylome controls which immune genes are active, determining immune responses. Too much immune activation equals autoimmune disease. Too little immune activation equals weak immunity. B cells, T cells, and natural killer cells are the frontline immune defenders. BH4 and NOS controls proper immune activation and regulation, directly impacting inflammation. Lymph nodes and spleen are immune training centers, and oxidative stress and mitochondria are the energy and immune efficiency. It must be kept in check or it will affect the integrity of your immune cells. The methylome controls immune system gene expression. Make sure that your immune responses are balanced, and the microbiome trains and regulates the entire immune system, and it acts as the immune system's trainer. So balance is key. If any part is dysfunctional, the immune system breaks down, leading to infections, inflammation, autoimmune diseases. What all these factors are to be in balance, the immune system is strong, controlled, and effective. When one is off, it can really mess you up and lead to chronic disease, autoimmunity, and infections. Now, I know I'm getting a little repetitive in here, but this is the stuff you need to understand because it's showing you how everything is all interconnected, and just one of these bullets could possibly unlock your whole healing journey for you. I know I just said I'm getting a little repetitive, and I apologize. Probably should have written this section a little bit better, but I didn't, and I apologize. This one is talking specifically about the methylome. So the methylome in T cells are controlling immune activation. T cells and proper DNA methylation to regulate their activation and avoid overreacting. If DNA methylation is too high, hypermethylation, T cells can become sluggish, leading to weaker immune responses and increased infections. If DNA methylation is too low, or hypomethylation, T cells may become hyperactive, leading to autoimmune diseases where they attack the body's own cells. The methylome can control memory T cells, ensuring they remember the past infections properly and responds faster the next time. The methylome in B cells, the antibodies production and autoimmunity. B cells rely on proper DNA methylation to differentiate into either plasma cells, which are the antibody factories, or memory B cells, which are the long-term immunity. Dysregulated methylation can cause B cells to overproduce antibodies, leading to autoimmune diseases like lupus, rheumatoid arthritis, and multiple sclerosis. If B cell methylation is impaired, they may fail to mature properly, weakening the body's ability to fight infections. The methylome and BH4, the regulating immune metabolism. BH4 is essential for controlling immune cell energy and function. DNA methylation controls genes involved with BH4 production. If methylation is off, BH4 levels can become too low, leading to weaker T and B cell responses and higher oxidative stress. Low BH4 equals increased immune dysfunction and mitochondrial stress and chronic inflammation. The methylome and NOS, which is your nitric oxide, and that balances inflammation. DNA methylation regulates the NOS enzymes, which produces nitric oxide. If the NOS genes are over-methylated, nitric oxide levels can be too low, reducing the immune system's ability to fight infections. If NOS genes are under-methylated, too much of nitric oxide can be produced, leading to chronic inflammation, mitochondrial damage, and immune overactivation. So as you can see, methylation, mitochondria, and your gut microbiome are essential for your immune system working together and properly. Now there's a lot of folks out there that are saying that methylation doesn't have that big of a play in your health, but it so does. So please understand just how important methylation is. And if you have the MTHFRs, this is going to hamper your methylation. So you have to be more careful. Last slide, everyone. Yay! The methylome in lymph nodes and spleen, controlling immune cell activation. So we are still talking about the methylome. The methylome helps regulate immune cell gene expression in the lymph nodes and spleen, where T and B cells are activated. Poor methylation can cause miscommunication between immune cells, leading to either a weak immune response or an overactive immune response. And remember, your methylome is part of your microbiome. So the methylome in mitochondria, the root of oxidative stress. DNA methylation controls mitochondrial function, which affects how much energy immune cells have to have in order to fight infections. If methylation is off, mitochondria produces too many free radicals, leading to oxidative stress, damaging your T and B cells and causing immune exhaustion. Methylation also affects genes involved in the glutathione production, and that is your body's master detoxifying biochemical. And it is a key antioxidant that protects mitochondria from oxidative stress. So, the immune system and methylation connection. Proper methylation keeps immune cells balanced. Not weak, not too aggressive. Dysregulated methylation can lead to immune dysfunction, increasing risks of infection, inflammation, and autoimmune diseases. Methylation affects BH4, NOS, mitochondria, lymph nodes, and spleen, which all directly impacts how T and B cells function. Fixing methylation can help restore immune balance, as well as your microbiome, because remember, the methylome is in your microbiome, and this will help reduce inflammation, improve energy, and support long-term immunity. And this is often why I'm always talking about fasting, because it stops all of these aberrant processes from running amok. It also helps all of these systems work efficiently together. Now, there's a lot of talk out there that the MTHFRs are not important and it doesn't affect your overall health. And this is all part of methylation. And I'm here to tell you that that simply isn't true. If they're saying that, it is because they don't understand the intricate relationship between everything, because the microbiome, the methylation, your mitochondria, it is so intertwined, it's not even funny. So if you hear someone telling you that the MTHFRs and methylation are not important, go find another doctor. Or educate yourself so that you understand how this relationship is tied together. Because without one, the other doesn't work. And as you can see, as I have pinpointed in these slides, you need them to work properly in order for the whole cycle of illness to work properly so that you can get out of chronic illness. It's also, if it's one area is not working properly, it messes with the entire cycle of illness and it sends a domino effect or a cascade effect, sending you into chronic illness. I have done my absolute best to try to illustrate all of this and educate both patients and doctors alike so that they can understand how everything ties together, because medicine is compartmentalized. The body is not. All right, this concludes part 1 of the cycle of illness immune system. I had to divide part 1 into 2 sections because it is over 7 hours long. So stay tuned for part 2 where I go over the microbiome. And the microbiome is a much shorter section, but it's extremely chocked full of information to help you and your health so that you can understand how everything fits together. And part 3 ties it all together and brings it home for you, and it will explain with the conjunction of the entire cycle of illness series exactly what is happening in your body and bring the whole message home and unlock so much for you so that you can understand what is happening. So the immune system is made up of your immune cells, immune organs, and your microbiome. So it would be a great disservice if I did not tell you how the microbiome affects every other system in your body, because it is a linchpin for the entire cycle of illness and how it works. On top of that, it will explain everything, and you will have so many aha moments, it will be ridiculous. But remember, you have to put it together with all the symptoms you're having and the rest of the cycle of illness. Stay tuned for the next video, which will be the microbiome, and I will be getting that out to you as soon as possible. Until then, love and light, happy healing, and remember, you are not alone. This is why I'm doing all of this work, so that you don't feel alone. There are people out there doing their best to help you, and we may not have ever even met. This has been a labor of love, tears, and frustration, and oh, so many hours behind a computer. Over 10 years of scientific study. And I truly hope that this video series has helped you a great deal.